Hold your breath for a moment, just a few seconds, long enough to notice something. The instant you stop breathing, something in your body starts to slow down. Your heart rate drops a little, and it drops more if your face happens to be in cold water at the same time. You probably won't notice it on land, in a warm room on a dry evening. But if you're ever in cold water and you submerge your face, your pulse will slow by 10 or 20 beats a minute, sometimes more, within seconds. There's a name for this. It's called the mammalian diving reflex. And the reason it's called that, rather than the human diving reflex, is that you share it with every other mammal on Earth. A dog has it, a mouse has it, an infant has it more powerfully than you do, which is partly why a small child pulled from cold water after many minutes without air can sometimes be revived with no brain damage at all. The sperm whale has it too. The sperm whale's version of this reflex, scaled and extended across something like 50 million years of selection, is what allows it to fall from the surface of the ocean to depths of 2,000 meters in total darkness, hunt a giant squid by echoing sound off its body, and return to the air an hour and a half later. The physiology is not new. The mammalian plumbing underneath is the same plumbing in your chest now as you're listening to this. What changed for the whale lineage was the degree to which selection pushed the reflex. Longer dives, deeper water, a slower heart, a body re-engineered around the ability to hold its breath. This is the story tonight. 50 million years ago, in what is now northern Pakistan, a small carnivorous mammal about the size of a wolf was drinking at the edge of a shallow river. It had 4 legs, hooves on those legs, fur, and a skull that paleontologists would later recognize as the earliest member of an entirely new evolutionary experiment. Almost nothing about its body would have suggested what its descendants would become. It moved on land the way a wolf moves. It hunted the way a wolf hunts. But hidden inside its skull, in the bones surrounding the inner ear, there was already something different. A subtle re-architecture of the tympanic bulla, the bone that houses the hearing apparatus, toward a structure that in every later whale would be diagnostic of the entire lineage. The animal did not know it was a whale. Nobody looking at it would have called it one. But the ear had begun to change, and the ear was the first commitment. What this documentary is about is the slow, blind, 50 million year process by which that small land mammal's lineage produced the animals now swimming in every ocean on Earth. Not all at once, not in a line. In a branching, messy, often dead-ending bush of species, most of which left no descendants at all, with one branch, roughly, making it through. The fossils that record the transition are, collectively, one of the most thoroughly documented major evolutionary transitions in the vertebrate record. For most of the 20th century, the origin of whales was considered one of the hard problems in mammalian paleontology. Over the last 40 years, it has become one of the solved ones. The animals that will appear in this story, Pakicetus, Ambulocetus, Rodhocetus, Maiacetus, Peregocetus, Dorudon, Basilosaurus, and the modern whales their lineage eventually produced, are not a ladder. They are more like a series of frames lifted from a much larger reel. The reel is missing most of its film. What we have are moments: a skull, a pregnancy preserved in sediment, an ankle bone that, when it was finally examined, overturned a consensus that had held for 4 decades. These moments, assembled carefully, tell a story about a lineage of small hoofed mammals that walked into the ocean and stayed there, and about what was left behind inside those animals' bodies and inside yours of the land they came from. Before any of that, a small favor. If what you're listening to tonight feels worth being made, the subscribe button does more for a channel like this than almost anything else. And in the comments, I'd like to know something. The mammalian diving reflex I mentioned at the start, have you ever felt it? Cold water on your face, Your heart slowing in a way you couldn't consciously control? I read the answers, and I find that question more interesting than almost anything I could tell you about whales. Now, if you're comfortable, get a little more comfortable. Your jaw is probably tighter than it needs to be, and your breathing will lengthen on its own over the next few minutes. You don't have to help it. The story starts in a warm, shallow river basin in what is now Pakistan, about 50 million years ago, where a small predator with hooves was watching something move through the water. It did not know it was the ancestor of anything. It was at that moment just hungry. 66 million years ago, a rock the size of a small city struck the Yucatan Peninsula at something like 20 kilometers per second, and for about 1,000 years afterward, the Earth's oceans were effectively uninhabitable for most of the creatures that had lived in them. The impact vaporized roughly 150 cubic kilometers of limestone and gypsum, filling the upper atmosphere with sulfate aerosols that blocked sunlight across the globe. Marine photosynthesis collapsed within weeks. The plankton died, and then the animals that ate plankton died, and then the animals that ate those animals died, and the die-off moved up the marine food chain with the grim, methodical efficiency of a thing being unbuilt from its foundation. By the time the sun returned and the oceans began to warm again, something like 75% of all marine species had disappeared, and among the casualties were almost every large marine predator the Mesozoic had produced. The mosasaurs were gone. 15-meter serpent lizards that had patrolled the shallow seas of the Cretaceous, cousin to modern monitor lizards but built entirely for the water, went extinct in full. The plesiosaurs, the long-necked reptiles with their strange paddle wings and dinner-plate heads, went with them. Large sharks were decimated, though they would eventually recover. The great Cretaceous marine reptiles, the animals that had filled the role of top predator in the oceans for over 100 million years, vanished more or less simultaneously, and what they left behind is what matters for everything that comes after. They left behind an empty ocean, not empty of life. Life recovered. It always does. Within a few million years, the surviving small marine reptiles, the recovering sharks, the first hints of what would eventually become modern bony fish, the early lineages of what would become seabirds, all began filling the middle parts of the marine food web. But the top part, the apex predator role, the large-bodied, fast-swimming niche at the summit of the ecological pyramid, that part stayed open. For roughly 15 million years after the end of the Cretaceous, no tetrapod predator larger than a modern seal occupied the deep oceans. This was a historically unusual situation. For most of the preceding 300 million years, something big had always been there. And now, for the first time in a geologically long while, nothing was. An empty niche is not a neutral thing in evolutionary terms, it is a pressure gradient. A patch of ocean with abundant fish, abundant cephalopods, abundant small marine mammals, and no large predator above them is a patch of ocean that will, sooner or later, select for whatever lineage happens to start moving into it. The lineage doesn't need to be well suited to begin with. It needs to be slightly better suited than its immediate relatives for long enough in the right generations. Selection does the rest. Something worth sitting with for a moment. The mammals who would eventually fill this niche had been present throughout the entire reign of the marine reptiles and had not moved into the ocean at all. For over 100 million years of the Mesozoic, mammals were small, nocturnal, insectivorous creatures about the size of shrews and rats, and the oceans were closed to them because the oceans already had tenants. It wasn't that mammals lacked the physiological potential to return to the water. Some lineages of modern mammals, otters, seals, hippopotamuses, would eventually demonstrate that the potential was always there. It was that the door had been locked by incumbents who were good at their jobs, and the door stayed locked until those incumbents died. What looks, in retrospect, like an inevitable evolutionary march of mammals into the sea, was actually contingent on a very specific astronomical accident occurring exactly when it did. Your four-chambered heart is the relevant organ to think about here. The same heart you have in your chest right now, two atria, two ventricles, valves between them, a muscular pump capable of sustained aerobic output, is the heart every mammal has. It is a design, if I can use that word carefully, that evolved in a distant common ancestor of all mammals during the late Permian and early Triassic, about 250 million years ago. It is built for endurance rather than short bursts, for maintaining oxygen delivery across long periods of activity, for the kind of sustained running or swimming that a cold-blooded reptilian heart cannot easily produce. When mammals finally did enter the oceans, they did not have to evolve a new kind of heart. They already had one. The same organ in your chest, scaled up across 50 million years and factors of hundreds in body size, is what eventually pumped blood through a blue whale. Nothing about the architecture changed, only the dimensions. This matters because it frames a common misunderstanding about what the whales did. The popular version of the story sometimes suggests that the mammal-to-whale transition required some extraordinary reinvention of the mammalian body plan, as if the lineage had to construct new organs from scratch to survive in water. The opposite is closer to the truth. Almost everything a modern whale has, every major organ system, was inherited from its land-dwelling ancestors, and the transition consisted mostly of reshaping what was already there, rearranging the limbs, migrating the nostrils, reinforcing the ear, modifying the circulatory pattern for prolonged breath-holding, extending reflexes already present in every mammal. The whales did not leave their mammalian body behind. They took it with them. It is worth naming the teleological trap here, because this is exactly the kind of story that invites it. It is tempting to say that the mammals took advantage of the empty ocean, or that a particular lineage chose to move toward the water, or that evolution found a way to fill the vacant niche. All of these formulations put intention into a process that has none. What actually happened is simpler and stranger. In each generation of a lineage of small coastal mammals, some individuals happen to spend more time in shallow water than others. Those that spent more time in water in the particular ecological circumstances of the early Eocene happened to produce slightly more surviving offspring than those that spent less. Across enough generations, the average behavior of the lineage shifted toward the water. No choice was made, no advantage was taken. The water didn't call anybody. The lineage drifted under statistical pressure in the direction the numbers were pushing it, and the numbers were pushing it because the niche above was open and the niche it was currently occupying was competitive. This looks, from the outside, like a design solution, a clever evolutionary move, an elegant filling of an ecological gap. It is none of those things. It is the aggregate result of millions of individual organisms pursuing their own hunger and their own reproduction, for tens of thousands of generations, with no one at any point having any idea what the lineage was doing. The elegance is entirely in the outcome. The process is blind. The ocean, for about 15 million years after the Chicxulub impact, was a large opportunity with nothing pressing against it. Then, in a warm, shallow sea at the margins of what is now the Indian subcontinent, something started to press. In 1971, an Indian geologist named Ranga Rao was working through the Eocene rocks of the Calicut region in Kashmir, about 48 million years old by the best available dating, and he found some teeth and fragments of a jawbone belonging to an animal nobody had described before. He named it Indohyus, meaning Indian pig, which was a reasonable guess given the teeth. It looked like it belonged to some small deer-like or pig-like artiodactyl, one of the even-toed ungulates that had been diversifying across Asia throughout the early Eocene. Rao published a short description the following year and went on to other projects. The fossils were filed away. Indohyus became, for the next 3 decades, a footnote, one of hundreds of obscure early artiodactyls, known mostly from teeth, of interest to specialists and to essentially nobody else. When Rao died in the early 2000s, the rocks he had collected were still sitting in crates. His widow gave the crates to an American paleontologist named Hans Thewissen, who had been working on early whale evolution in Pakistan and who she thought might find the material useful. Thewissen had been interested for some time in a question that had been nagging at whale paleontology. The molecular evidence from the 1990s, the mitochondrial DNA comparisons, the nuclear gene analyses, and eventually the sine retrotransposon insertion data had all been pointing toward a close relationship between whales and the hippopotamus family. If that was right, then whales were not just related to artiodactyls but nested inside them, cousins to hippos and more distant cousins to deer, pigs, and cattle. The problem was morphological. No known artiodactyl fossil looked anything like a whale. The oldest whales, the Pakicetus, were recognizable as cetaceans by the structure of their inner ear and by certain features of their teeth. But the animals they seemed to share those teeth with in the fossil record were mesonychids, the extinct wolf-like ungulates that paleontologists had been proposing as whale ancestors for decades. There was, in other words, a sister group-shaped hole in the fossil record. Whatever artiodactyl had been the closest relative of the earliest whales was missing, and the molecular biologists knew it had to exist, and the paleontologists couldn't find it. One afternoon in 2006, a technician in Thewissen's lab was preparing one of the Kashmiri specimens, carefully removing the surrounding matrix from a small partial skull of Indohyus, and the skull broke. It was an accident, the kind of thing that happens in paleontology labs, usually met with a quiet curse and an hour of repair work. But when Thewissen looked at the broken surface, he saw something he was not expecting. The inner ear region was exposed, and the auditory bulla, the bony housing that surrounds the middle ear, had a particular shape and thickness that he recognized instantly. The bulla was formed entirely from the ectotympanic bone, not from the multiple bones that contribute to the bulla in other mammals. It was thickened, dense, pachyostotic. It was, in its architecture, the same feature that diagnosed every whale in the fossil record from Pakicetus onward. Nothing else had this structure. Not any artiodactyl, not any other mammal on Earth. Only cetaceans. And now this small deer-like creature from Kashmir. Thewissen spent the next year checking and rechecking the find, assembling a team, running the isotopic analyses, looking at bone density, comparing the dentition to Pakicetid teeth, building the phylogenetic tree. The paper appeared in Nature at the end of 2007. Indohyus, the animal the world had known for 36 years as an obscure little artiodactyl, was the sister group of whales. The missing morphological link between the hippo whale clade and the rest of the artiodactyls had been sitting in a crate for decades, and it had taken a broken skull to reveal it. What Indohyus tells us, beyond settling the phylogenetic puzzle, is something about what the earliest stages of the return to water actually looked like, because Indohyus was not a whale. It was not even close to being a whale. It was a small herbivore, about the size of a house cat, with 4 legs, hooves, a long tail, and a body plan appropriate for moving around on land. But its bones were dense, pachyosteosclerotic, to use the technical word, the same kind of heavy, marrow-reduced skeleton that modern aquatic mammals like hippos and sea cows carry. Dense bones work as ballast in water. They make it easier to sink, easier to stay submerged, harder to be swept up by a current. The oxygen isotope ratios preserved in Indohyus's teeth confirm what the bones suggest. This animal was spending a lot of its time in water. Not hunting in water, because its teeth are clearly those of a herbivore, but using the water for something else. The likely explanation comes from a living animal that does essentially the same thing today. The African water chevrotain, sometimes called a mouse deer, is a small forest-dwelling herbivore from West and Central Africa that, when threatened by a predator, runs into the nearest body of water and submerges. It can stay under the surface, holding its breath, for up to 4 minutes. It walks along the bottom. It waits for the predator to lose interest, and then it surfaces, shakes itself off, and resumes feeding. This is not an aquatic lifestyle in any meaningful sense. The chevrotain is not a swimmer or a diver. It is a land animal that uses water as a hiding place. Thewissen and his co-authors argued that Indohyus was probably doing something very similar, living as a small terrestrial herbivore on the margins of the Tethyan wetlands, fleeing into shallow water when pressed by predators, and over many generations evolving denser bones because the individuals who could stay submerged longer without bobbing back to the surface in panic survived to reproduce more often than those who couldn't. Think about what this implies. The ancestor that the whale lineage shares with Indohyus was not being selected for swimming efficiency, for diving physiology, for any kind of oceanic adaptation. It was being selected for the ability to hide in water when a predator showed up. The initial commitment to the aquatic environment, in other words, was driven not by the ocean's rewards, but by the land's dangers. Long before any of this lineage's descendants had any interest in fish or squid, or the deep pelagic niches that would eventually reshape their entire bodies, their ancestors were already spending time in water for a completely different reason. They were afraid. That's what got them wet. Everything that came afterward, every anatomical change, every behavioral shift, every reconstructed organ system, was built on the foundation of a reflex that was originally about escape. Your own body has a version of this. If you've ever been startled while standing near a pool or fallen unexpectedly into cold water, you'll know the specific physiological response. The sharp intake of breath, the instant rerouting of attention to the water itself, the odd combination of panic and involuntary focus that comes with sudden immersion. It's the older mammalian reflex, still intact, still running on the same neural circuitry that drove a small Kashmiri herbivore into a shallow pond 48 million years ago. The circumstances that shaped your ancestors' relationship with water have been very different. Your lineage went the other way, towards savannas and forests and eventually cities. But the same ancient mammalian toolkit is what makes you gasp when you fall in. There is one more detail about Indohyus worth sitting with because it quietly illustrates how evolution actually works. The dense, ballast-like bones that allowed Indohyus to stay submerged look, at first glance, like exactly the kind of adaptation that a designer might have given an animal preparing for life in water. They appear purposeful. They appear solved. But pachyosteosclerotic bone is not a feature that evolution produces deliberately. It is what happens when certain normal bone remodeling pathways are suppressed, when the ordinary process by which bones hollow themselves out for lightness simply doesn't run. The heaviness isn't built. It's what you get when the lightening stops. Hippos have the same trait, arrived at independently. Sea cows have it. Certain extinct marine reptiles had it. Each time, what looks like a design for sinking is actually the signature of a developmental program that was allowed to drift away from the default mammalian pattern because, in that particular environment, the drift produced a survival advantage. Nothing was designed. Something stopped being maintained, and the stopping turned out to be useful. That's the shape of most evolutionary adaptation, when you look at it closely. Not the construction of new features, but the modulation of old ones. Not invention, but the loosening or tightening of constraints already present in the genome. Indohyus did not evolve heavy bones for water. Water selected over many generations for the individuals whose ordinary bone development happened to produce extra density, and the rest of the lineage followed the numbers. The ballast is a side effect of a developmental program that was pushed by selection a little past its usual settings. The animal that emerged, small, slow, densely boned, spending hours a day in shallow water hiding from whatever wanted to eat it, was not yet a whale. It was not going to become a whale in any individual generation. But the lineage it belonged to was, at this moment, on a trajectory that no other mammal lineage had ever been on before. The trajectory had not yet committed to the ocean, it had committed to the water's edge. Somewhere nearby, in the same wetlands, another lineage was about to commit to something stronger. The rocks of the Kuldana Formation in northern Pakistan are a reddish-brown sandstone that weathers into rounded hillsides and shallow gullies along the edge of the Himalayan foothills. 50 million years ago, before the collision between the Indian plate and the Asian landmass had finished throwing those mountains into the sky, this area was the coast of a different kind of world. The Tethys Sea, which no longer exists, washed against shorelines roughly where central Pakistan now sits, and the landscape behind those shorelines was warm, low-lying, heavily vegetated, and laced with rivers that ran slow and shallow through alluvial flats. It was the kind of environment where crocodilians would have been common, where turtles would have sunned themselves on mudbanks, where early horses about the size of small dogs would have moved through the undergrowth. It was also, though nobody walking the ground at the time would have had any way of knowing it, the birthplace of the whale lineage. The first Pakicetus fossils came out of these rocks in the late 1970s, when a team led by Philip Gingerich of the University of Michigan was working through the Eocene deposits of the Kohat district. What they found initially was fragmentary: a partial skull, a broken mandible with some teeth still in place, pieces of the skeleton that didn't obviously go together. Gingerich and his colleague Donald Russell published a short paper in 1981 describing the animal and naming it Pakicetus inachus. They thought, based on the teeth, that they might be looking at a masonykid, one of the extinct wolf-like ungulates that paleontologists at the time believed were the likely ancestors of whales. The teeth had the right general shape. The skull, from what little of it they had, didn't immediately suggest anything else. For about a year, Pakicetus was just another Eocene mesonykid, notable mainly for being unusually well preserved. Then somebody looked at the ear. Specifically, they looked at the underside of the skull, at the bony housing that surrounds the middle ear chamber. In mammals, this housing, the auditory bulla, is usually made up of contributions from several different bones fused together in species-specific ways. Mesonychids had a bulla built the normal mammalian way. But the bulla of this Pakicetus skull was different. It was formed entirely from a single bone, the ectotympanic. It was thickened to the point of being almost absurdly dense, filled with marrow that was compressed into something closer to solid stone than normal bone tissue. And its shape had a particular curvature that, as far as anyone knew at that point, existed in exactly one group of animals: whales. Every whale that had ever been described from the Basilosaurids onward had this same structure. Nothing else did. The feature was called the involucrum, and it was considered, at the time and still today, the single most diagnostic bony signature of the cetacean lineage. This put Gingerich and Russell in an unusual position. They had an animal that looked from the outside like a wolf-sized terrestrial predator with hooves and a long tail and 4 fully functional legs. Its teeth, its limb bones, its general body plan. All of it said land mammal. But the part of its skull that most reliably identifies an animal as a whale said, unambiguously, whale. They revised the paper. Pakicetus was not a mesonychid, it was a cetacean, the oldest cetacean anyone had ever found. It is worth pausing here to appreciate how strange a conclusion this was at the time. The Pakicetus skeleton, when more of it was eventually recovered in the early 2000s, made the strangeness even starker. This was an animal with cursorial limbs, meaning limbs built for running, with rigid elbow and ankle joints, long, slender bones, proportions characteristic of mammals that chase prey across open ground rather than swim. Its shoulder blades had large attachment sites for running muscles. Its pelvis looked like the pelvis of a land predator. Its feet were not webbed. Nothing about its body below the neck suggested any kind of aquatic lifestyle. The team that published the postcranial description in 2001, again Thewissen and colleagues, wrote that Pakicetus was, in their words, no more amphibious than a tapir, which is to say, not very. And yet, the ear. The ear said whale, and the ear doesn't lie. What the Pakicetus ear tells us, if we read it carefully, is something about the order in which the return to water actually happened. Most of us, I think, imagine evolutionary transitions as a kind of coordinated project. The limbs changing at roughly the same time as the physiology, the behavior, the body shape, everything moving together in a consistent direction. Real transitions don't work that way. They tend to start with one specific structure, one narrow functional change, and the rest of the body follows slowly over millions of years, often lagging behind by tens of millions of years in some systems. In whales, the thing that changed first was the ear, not the legs, not the nose, not the lungs. The ear. The lineage that would eventually become every whale on Earth committed first to the acoustic properties of water and kept almost everything else terrestrial for several million years afterward. The reason this order matters is that it tells us what the original selective pressure probably was. Pakicetus was not swimming out into open water hunting fish. Its body couldn't have done that. The cursorial skeleton rules it out. But it was probably standing in shallow water at the edges of those slow Tethyan rivers, listening, waiting. Animals came to the water to drink. Fish moved through the shallows. Small mammals crossed at fords. And any predator that could hear what was happening in the water itself, pick up the vibrations and low-frequency sounds traveling through liquid rather than air, would have had an advantage over competitors that could only hear what was above the surface. The ear began to modify because hearing underwater, even slightly better than baseline, turned into more meals and more offspring. The rest of the body hadn't yet had any reason to change. There's a small mechanical problem worth understanding about underwater hearing in mammals, because it explains why the ear had to be rebuilt rather than just extended. A normal mammalian ear, the kind you have in your own head, depends on sound waves passing through air, hitting the eardrum and vibrating the three small bones of the middle ear, the malleus, incus and stapes, which amplify the vibration and pass it into the fluid-filled inner ear, where the cochlea translates it into neural signals. This system works beautifully in air. In water, it collapses. Water is about 800 times denser than air, and when the whole head is submerged, sound doesn't so much enter the ear canal as pass through the entire skull, arriving at both inner ears simultaneously and with nearly identical intensities. A terrestrial mammal underwater can hear that something is making noise, but it cannot tell which direction the noise is coming from. And for a predator, directional hearing is not optional, it is the entire point. What the Pakicetus ear was beginning to do was acoustically isolate the middle ear from the rest of the skull. The pachystotic thickening of the ectotympanic bulla, the involucrum's particular geometry, the sinuses of air that would eventually form around the ear in later whales, all of these were selected for one reason, which was to stop sound from reaching the inner ear through the bones of the skull. Once the inner ear is isolated, incoming sound has to go through the properly mammalian route, and the tiny differences in arrival time between the left and right ears become detectable again. Directional hearing becomes possible. The predator can tell where the fish is. Your own 3 middle ear bones, the malleus and incus and stapes, are the same 3 bones Pakicetus had. They are, in an evolutionary sense, quite old. They came from the jaw bones of our reptilian ancestors, reorganized during the early evolution of mammals about 200 million years ago into a sound-transmitting chain that would define mammalian hearing from that point onward. When the whale lineage started adapting these bones for underwater use, they weren't inventing anything new. They were modifying equipment that had already been modified once before from a completely different original function. The jawbones of a reptile became the hearing bones of a mammal, became, eventually, the acoustic isolation machinery of a whale. Each step used what the previous step had left behind. Nothing started from scratch. Nothing ever does. The Pakicetus skull, when you see it in a museum or in a high-quality reconstruction, does not look impressive. It looks like a wolf's skull with a slightly oversized ear hole. The creature it belonged to, judging by everything we now know about its post-cranial anatomy, probably looked a lot like a mid-sized fox or small coyote, maybe with slightly longer legs, maybe with a somewhat more elongated snout. If you had encountered a living Pakicetus in the Pakistani scrubland of the early Eocene, you would not have called it a whale. You would not have thought it had anything to do with the ocean. You might not have given it a second look. And yet, that small predator was already, at the level of anatomy that mattered, committed. One bone in its skull had crossed a line the rest of the lineage would eventually have to follow. The line, for a few million years, was very thin. Imagine a river delta in what is now northern Pakistan 49 million years ago. The water is warm and brackish, a mixture of fresh water running down from inland and salt water pushing in from the Tethys at high tide. Mangrove-like vegetation grows along the banks, and between the tangled roots, the bottom of the river is a soft mud a meter deep in places. Small fish move through the shallows in darting schools. Turtles sun themselves on exposed logs. Herds of small early mammals, dichobunids, anthracotheres, the first ancestors of horses, come down to the water in the late afternoon to drink, and they come carefully because something about the surface of the water has been bothering them lately. The ripples are wrong. The shadows under the mangroves move when they shouldn't. What they are sensing, without being able to articulate it, is Ambulocetus natans, a 4-meter predator, roughly the length of a modern saltwater crocodile and with a similar posture, long body, short limbs, powerful tail, heavy jaws. It lies motionless in the shallows, with only the tops of its eyes and the tips of its nostrils breaking the surface, a position it can hold for hours. Its eyes are set high on its skull, rotated upward the way a frog's eyes are, so that it can watch the bank above the waterline while the rest of its body stays hidden. When something edible comes within range, Ambulocetus explodes out of the water in a single lunge, grabs the prey with a bite force strong enough to crush ribs, drags it back under, and holds it there until it drowns. Then it eats. This animal was a whale. It is worth sitting with that claim for a moment because it sounds wrong. Ambulocetus did not have flippers. It did not have a blowhole. It did not swim the way modern whales swim. It had 4 fully functional legs, shorter than Pakicetus's, but still adequate for moving on land, and it probably spent a meaningful fraction of its time out of the water, hauling itself onto banks to rest, perhaps to sleep, possibly to give birth. Its body was not streamlined. Its tail, though powerful, did not have a fluke. Everything about it that a casual observer would notice was more reptilian than cetacean, more crocodile than whale. And yet, the ear was there. The involucrum was fully developed. The dense, acoustically isolated bulla that had just begun to appear in Pakicetus was, in Ambulocetus, completely formed. This animal could hear underwater with the kind of directional precision that a terrestrial predator simply cannot achieve. That ability, and the body built around exploiting it, is what makes Ambulocetus a whale, even though nothing about its silhouette suggests it. The first fossils of Ambulocetus came out of the Kuldana Formation in the early 1990s, a few kilometers from where the earlier Pakicetus specimens had been found. Thewissen and his colleagues, Syed Taseer Hussain and Muhammad Arif, published the initial description in Science in 1994, calling the animal Ambulocetus natans, which means, roughly, the swimming, walking whale, a name that tries to capture the transitional quality of the body in a single binomial. Over the next several years, continued excavation at the type locality recovered about 80% of the skeleton. By the time Madar, Thewissen and Hussain published their detailed 2002 analysis, Ambulocetus had become one of the best-known early whales in the fossil record and one of the strangest. The strangeness is in the locomotion. Ambulocetus could not have swum the way modern whales swim. Modern whales propel themselves with their tails, moving the flukes up and down in a powerful vertical stroke that uses the muscles of the lower back. Ambulocetus had no flukes, and its tail, while muscular, was not built for that kind of propulsion. What it had instead was a pelvis still firmly articulated with its spine, hind limbs still capable of bearing weight, and a spinal column flexible enough to undulate in the same dorsoventral plane that mammals use when they run. The best reconstruction of Ambulocetus's swimming is that it moved a little like a modern otter, flexing its back up and down, kicking with its hind legs, using its broad webbed feet as paddles. This is not a whale's swimming, it is a mammal's swimming, the kind any moderately athletic land mammal can do, extended and optimized. But the undulation of the spine, the dorsoventral flex of the lower back, that was the seed. In modern whales, the same motion drives the tail fluke. The muscles and the skeleton required for the whale's characteristic propulsion were already there in Ambulocetus, doing a different job. Nothing new had to evolve. The mammalian running gait, which is itself a dorsoventral flex of the spine, was repurposed into a swimming stroke, and then, over the next 15 million years, the hind legs shrank, the tail grew flukes, and the same basic motion became the signature locomotion of every whale now alive. If you watch a dolphin swim, what you are watching is a galloping mammal that never stops galloping, its back arching and releasing in a rhythm that a fox or a cheetah would recognize. The hunting strategy of Ambulocetus is where this chapter's real interest lies, because it illustrates a principle about evolutionary convergence that the whale lineage will repeat at larger and larger scales as it moves forward through time. The niche Ambulocetus occupied, large aquatic ambush predator at the water's edge, grabbing terrestrial prey that came to drink, was not a new niche in the early Eocene. Crocodilians had been occupying it for something like 150 million years already. Mosasaurs had occupied versions of it in the Cretaceous. Phytosaurs had occupied it in the Triassic. Eogerinids and related early tetrapods had occupied proto-versions of it in the Carboniferous. The ambush predator at the water's edge role is one of the most stable ecological roles in vertebrate history, and essentially every time a major tetrapod lineage has become semi-aquatic, some branch of that lineage has ended up in the ambush predator slot. What's striking is that when unrelated lineages converge on this niche, they tend to converge on the same body plan. Long body, short limbs, laterally flattened tail, jaws at the front of a low, flat skull, eyes rotated upward, nostrils at the tip of the snout so the animal can breathe while mostly submerged. This is the crocodile shape, but it is also the phytosaur shape, and the early tetrapod ambush predator shape, and, for a few million years in the early Eocene, the whale shape. Ambulocetus is a mammal that evolved under the selective pressures of a particular kind of aquatic ambush lifestyle into something that looked remarkably like a crocodile. It was not a crocodile. It shared no recent common ancestor with crocodiles. It arrived at the crocodile body plan from an entirely different starting point through an entirely different evolutionary pathway because the physics of ambushing terrestrial prey from shallow water selects for the same solutions regardless of what lineage is doing the evolving. This pattern, the same body plan recurring independently in unrelated lineages, is what evolutionary biologists call convergent evolution. And it is one of the most persuasive things in the natural world. Every time you encounter it, you are watching something remarkable. The demonstration that body shape, under sustained selective pressure, is not arbitrary. There is a set of physical problems that an ambush predator in shallow water has to solve: concealment, explosive acceleration over short distances, the ability to hold prey underwater, the ability to breathe while mostly submerged, and the number of workable solutions to those problems is small. Lineages that try other solutions die. Lineages that arrive at the crocodile shape, or something close to it, survive. Across tens of millions of years and dozens of unrelated evolutionary experiments, the same shape keeps emerging, not because any designer prefers it, but because the alternatives don't work. This is a version of the Dawkins argument about the eye. Eyes have evolved independently at least 40 times across animal evolution, in cephalopods and vertebrates and arthropods and jellyfish and flatworms, and every time they have evolved, they have arrived at a small number of recurring designs: camera eyes, compound eyes, pit eyes, because the physics of light and the biology of detecting it permit only a limited number of workable arrangements. Ambush predator body plans are the same kind of phenomenon. The shape looks designed because the constraints are real and the options are few, but nothing designed it. 40 different lineages, or in this case at least a dozen, solved the same problem by stumbling onto the same solution, and the recurring solution is what we now call the crocodile shape, because crocodiles happen to keep it longest. Your own mammalian jaw, the one you are chewing with when you eat, is the articulated two-part system that all mammals inherit from a specific group of reptilian ancestors in the late Permian. It is a very good jaw for a terrestrial mammal. When the whale lineage ran into the ambush predator selective regime, the same jaw was modified, reinforced, elongated, repositioned on the skull to do the work that a crocodile's jaw does. A different kind of jaw would not have evolved into the crocodile shape as easily. The mammalian jaw, with its particular muscle attachments and its particular geometry, lent itself to this transformation because the transformation only required modifications to something the lineage already had. Ambulocetus held this niche for several million years. Then the lineage began to drift away from it, because the river delta ambush role, successful as it is, does not scale. The rewards of being an ambush predator at a water's edge are capped by the rate at which prey comes to the water. Out in the open ocean, the prey does not wait for you to come to it. It is there constantly, in enormous quantities, and the lineage that could leave the mangroves behind and start hunting in open water had access to a food supply essentially without upper limit. The animals that made that move were not descendants of Ambulocetus directly. They were descendants of one of its cousins. The cousins were already at the coast. For most of the 20th century, the question of where whales came from had a reasonably confident answer, and that answer was wrong. From roughly the 1960s through the late 1990s, the textbook Consensus, the one that appeared in undergraduate evolutionary biology courses, in museum exhibits, in popular books about whales, in the standard references that working paleontologists cited, held that cetaceans descended from an extinct group of hoofed carnivorous mammals called mesonychids. The mesonychids were a real and well-studied lineage, mostly Paleocene and Eocene in age, ranging in size from house cats to small bears, and some of them were genuinely impressive. Andrewsarchus, the largest known mesonychid, had a skull nearly a meter long and was arguably the biggest terrestrial carnivorous mammal that ever lived. These were wolves with hooves, essentially, predators that ran down prey on land and had dentition adapted for slicing meat from bone. The case for mesonychid ancestry of whales was built on teeth. Early cetaceans and mesonychids had broadly similar dental patterns, triangular cusped cheek teeth with a specific arrangement of crests that appeared, at a glance, to be shared. Given that teeth are overwhelmingly the most common thing to preserve in the mammalian fossil record, and given that the earliest whales known at the time were all represented primarily by teeth and jaw fragments, the mesonychid hypothesis was the inference the evidence seemed to require. Paleontologists like Lee Van Valen proposed it formally in the 1960s, and over the following decades it became solidified by the work of researchers including Maureen O'Leary, Jonathan Geisler, Zhexi Luo, and, notably, Philip Gingerich himself, who spent much of his early career operating within the mesonychid ancestor framework. Meanwhile, in a different branch of biology, a different answer was emerging. Molecular biologists in the 1990s had been using new sequencing techniques to compare the DNA of modern mammals and construct phylogenetic trees based not on shared anatomical features but on shared genetic sequences. When they did this for cetaceans, the result was consistent and, for the traditional paleontological community, surprising. Whales didn't cluster near mesonychids on these molecular trees. Mesonychids weren't present on the trees at all because they were extinct and had left no DNA to sample. What the molecular data showed instead was that whales were nested deep inside the artiodactyls, the even-toed ungulates, the group that includes cattle, sheep, deer, pigs, camels, and most relevantly for this story, hippopotamuses. Every independent line of molecular evidence pointed to hippos as the closest living relatives of whales. Mitochondrial cytochrome B sequences pointed there. Nuclear gene comparisons pointed there. Whole mitochondrial genomes pointed there. And then, in 1999, Norihiro Okada's group in Japan published a study using short interspersed repetitive elements, signs, which are essentially retrotransposon insertions that leave nearly unambiguous genetic signatures, and showed that the signy insertion patterns definitively placed cetaceans inside the artiodactyl clade as sister to hippos. This created a genuinely uncomfortable situation. Two entirely independent lines of evidence, both rigorous, both internally consistent, were giving incompatible answers. The morphological evidence from fossils said whales came from mesonychids, which are not artiodactyls. The molecular evidence from living species said whales are nested inside artiodactyls, sister to hippos. Both can't be right. Somebody had to be wrong. For a few years in the late 1990s, the situation was essentially a standoff. Paleontologists pointed out, reasonably, that molecular data can only sample living species, and living species represent a tiny slice of evolutionary history. Extinct lineages like mesonychids might be the actual ancestors, even if no molecular evidence survives to show it. Molecular biologists pointed out, also reasonably, that morphological similarity can be produced by convergent evolution, and the apparent dental resemblance between mesonychids and early whales might be a case of two unrelated lineages evolving similar teeth under similar selective pressures. Neither side could definitively refute the other. What everyone agreed on was that something had to give, and the way it was going to give was with a new fossil. specifically an early whale preserved well enough to show parts of the skeleton besides the teeth, parts that could test the mesonychid hypothesis and the hippo hypothesis against each other directly. The fossil that did it came out of Balochistan province, Pakistan, in the year 2000. Philip Gingerich's team was working in a different part of the country from where the Pakicetus and Ambulocetus specimens had been found, searching through Middle Eocene marine deposits for whales that were already committed to the water. What they recovered was a nearly complete skeleton of a new species they would name Rodhocetus balochistanensis, along with a related specimen called Artiodactyls clavis. These were protocetids, the evolutionary stage after Ambulocetus, more aquatic, smaller limbs, streamlined bodies, the beginning of what a modern observer would actually recognize as whale-shaped. But the thing that mattered about these skeletons was a single small bone in the ankle, the astragalus. The astragalus in mammals is the bone that sits between the shin and the foot, and it is one of the most diagnostic bones in the entire skeleton for questions of evolutionary relationship. Different mammal groups have distinctively different astragalus shapes, and the shape is strongly conserved. It doesn't change easily, because any change to the astragalus affects how the animal walks. Artiodactyls have an astragalus unlike any other mammal group. It has what paleontologists call a double pulley structure, two sets of grooved surfaces at either end of the bone, which together produce the particular hinging motion of an artiodactyl ankle. Every artiodactyl has this. Deer have it, pigs have it, camels have it, hippos have it, nothing else does. Not primates, not carnivorans, not rodents, not perissodactyls like horses, and critically, not mesonychids. The double pulley astragalus is a synapomorphy of the artiodactyl clade, a shared derived feature that defines the group. Rodhocetus had it. Gingerich, when he recognized what he was looking at, understood immediately what this meant. The astragalus had been the missing piece. For the mesonychid hypothesis to be right, early whales should have had the mesonychid ankle, a different shape entirely, without the double pulley. Instead, they had the classic artiodactyl ankle, indistinguishable in its basic architecture from the ankle of a deer or a hippo. The morphological evidence wasn't contradicting the molecular evidence anymore. It was confirming it. Whales were artiodactyls. They had always been artiodactyls. The teeth had misled everyone for 40 years because teeth, it turned out, can converge. Ankles are harder to fake. The paper came out in Science in September 2001, with Gingerich as lead author. In almost the same week, Thewissen's group published an independent analysis of Pakiceted and ichthyolestid ankle bones in Nature, showing the same double pulley astragalus in even older whales. Two teams, working separately, had arrived at the same conclusion from different specimens, and the conclusion was that the mesonychid hypothesis was dead. 4 decades of consensus dissolved in about 6 weeks. There is something worth noticing here about how science actually works in cases like this. Gingerich had been one of the most prominent advocates of the mesonychid hypothesis. He had published papers within that framework for 20 years. When the astragalus evidence came in, he wrote, in his later summary of the work, that the earlier idea that whales evolved from mesonychids was no longer tenable, and he included himself among those who had expected to find mesonychid affinities and didn't. He changed his mind in public on the basis of the new evidence, without hedging. This is not always how scientific arguments end. Sometimes the original advocates of a hypothesis dig in and die defending it, and the hypothesis survives them only to be quietly abandoned by the next generation. The whale ancestor question happened the other way. The people who had built the mesonychid framework were the ones who published the papers killing it, because the evidence in front of them no longer supported the position they had held. This is also a useful moment to sit with the larger question of what the whale-hippo relationship actually means. In evolutionary terms, hippos and whales share a common ancestor roughly 55 million years ago. Somewhere in the early Eocene, before the Pakicetus, before Indohyus, before any of the animals we've been tracking. The hippo lineage went in one direction, staying on land but developing a semi-aquatic lifestyle of its own, with dense bones and underwater birth and an ecological niche tied to African rivers. The whale lineage went in the other direction, into the ocean, and became what it became. The two lineages have been separated for more than 10 times as long as humans have been separated from chimpanzees. They look nothing alike. And yet, at the level of DNA, of genetic architecture, of ankle bones and ear bones, and the fundamental organization of the mammalian body, they are closer to each other than either is to any other living animal. Your own relationship to this clade is more distant, but not unrelated. Humans are placental mammals, like whales and hippos. The common ancestor we share with cetaceans and artiodactyls was a small mammal living sometime in the late Cretaceous or early Paleocene, probably shrew-like or rodent-like, running around in the undergrowth while the dinosaurs were still dominant or had just disappeared. From that common ancestor, our lineage went toward primates, toward apes, toward humans. The artiodactyl lineage went toward hooves, toward grazing, toward the even-toed ungulates. The cetacean lineage split off from the artiodactyl one and went toward the ocean. 3 major branches, diverging from a shared route, each unaware of the others, each pursuing its own local evolutionary logic. The ankle bone that settled the whale ancestor question is the same bone that you have in your own foot, modified by about 65 million years of primate-specific selection, but still recognizably the same structural element, still doing essentially the same job. The architecture is conserved because the underlying problem, attaching a foot to a leg in a way that allows directional locomotion, has essentially the same solution across most mammalian lineages. Small modifications produce large differences in gait. Large modifications destroy the animal. The whale lineage, by the time of Rodhocetus, had begun to make the large modifications. The double pulley astragalus was still there, a vestige of the terrestrial ancestry the lineage had spent millions of years walking away from. But the limbs around it were shrinking. The hind legs that had been powerful in Pakicetus, still functional in Ambulocetus, adequate in Rodhocetus, were on a trajectory that would end with nothing at all. The ankle bone would persist in reduced form all the way to Basilosaurus before finally disappearing from the external skeleton. Every modern whale, in its embryonic development, still briefly forms the beginnings of a hind limb, including what appears to be a tiny ankle, before reabsorbing the structure entirely. The genetic machinery for building an artiodactyl ankle is still there, still in the genome of every dolphin and sperm whale alive, still turning on briefly in the womb before being switched off again. The double pulley has been gone from adult whales for tens of millions of years, but the instructions for making one are still filed away, unused, waiting for a selective pressure that will never come. What Rodhocetus was doing, in the middle Eocene shallow seas off the coast of Pakistan, was swimming. Not well, by modern whale standards, but well enough. It had a body about 3 meters long, a long tail that was beginning to develop into something that could drive the animal through water, shortened limbs that could still bear weight on land but were no longer the primary locomotion system, and the full suite of inner ear modifications that would have let it hunt by underwater sound. It was an amphibian in the old loose sense, not a frog or salamander, but a creature genuinely at home in both environments, equally capable of hauling out onto a beach or swimming through open water. The ancestors of every whale alive today went through a stage that looked roughly like this for about 3 or 4 million years, and then the lineage kept moving. The coasts were full of them. The coast of southern Peru, a few hours' drive south of Lima, is one of the strangest landscapes on Earth. The Humboldt Current, flowing up from Antarctica along the western edge of South America, cools the air above the shoreline so thoroughly that almost no rain reaches the land, and the result is a desert that extends for hundreds of kilometers inland, pale, pebbled, almost entirely free of vegetation, broken only by occasional dry riverbeds and by the white bones of fossils weathering out of the rock. The Ica Valley, inland from the town of Pisco, is one of the richest marine fossil sites in the world. 40 million years ago, this was the sea floor. Today, it is a place where, if you know what you're looking for, you can walk across open ground and find the vertebra of a whale every few hundred meters, half buried in the sand, baked grey by the sun. In 2011, an international team of paleontologists working in this area, led by Olivier Lambert from the Royal Belgian Institute of Natural Sciences and including the Peruvian fossil hunter Mario Urbina, who had by that point spent decades combing the Ica rocks and had developed something close to a sixth sense for where bones might be, uncovered a partial skeleton at a site called Playa Media Luna. What they were looking at, as they began clearing away the matrix, was not immediately recognizable. There were ribs, vertebrae, parts of a pelvis, fragments of limb bones. The proportions were wrong for the common fossils of the Pisco Basin, which are mostly later Miocene sharks and whales that fit recognizable patterns. These bones belonged to something older, about 42.5 million years old, based on the stratigraphy, and the body plan they implied was something the Pacific Ocean had never, in all the work done on its fossil record, produced before. It was a quadrupedal whale. That phrase should land with some weight, because for the previous several decades, every quadrupedal whale known to paleontology, every whale with functional legs, every animal representing the transitional stage between terrestrial ancestors and modern fully aquatic cetaceans, had been found in a very specific part of the world: Pakistan and India, the Tethys Sea margins of the Indian subcontinent, in rocks between about 50 and 45 million years old. This was where the whale lineage had originated. This was where it had produced Pakicetus and Ambulocetus and Rodhocetus and all the other early forms, and this was where the quadrupedal chapter of cetacean evolution was assumed to have played out. Finding a four-legged whale in Peru, on the Pacific coast of South America, on the opposite side of the planet, meant that something had happened that the fossil record had not previously documented. Whales had crossed an ocean while still quadrupedal. They had done it before they had finished becoming whales. The team named the animal Peregocetus pacificus, which translates roughly as the traveling whale that reached the Pacific. They published the description in Current Biology in 2019, and the paper itself is worth reading if you can find it, because it works out the geographic puzzle with some care. The reasoning runs like this. The Peregocetus skeleton is clearly a protocetid, closely related to Rodhocetus and Maiacetus, the same evolutionary stage, the same general body plan. Protocetids of this type are otherwise known only from South Asia and from the coast of West Africa. Peregocetus's presence in Peru implies that the lineage dispersed from its Asian origin westward along North Africa, then across the Atlantic to South America, and then up the Pacific coast. The Atlantic was narrower then than it is now. The prevailing surface currents ran westward from Africa towards South America, the same way they still do today. A protocetid capable of sustained swimming, still able to come ashore to rest, could plausibly have ridden those currents across what was, at the time, a much shorter gap than the modern Atlantic, perhaps half the current width. The crossing would still have been extraordinary, but it was within the range of what a semi-aquatic whale in the Middle Eocene could have managed. Think about what this implies about the animal itself. Peregocetus was about 4 meters long, roughly the size of a modern sea lion, with 4 legs that were definitely still functional on land. The hoof-like structures on its toes make that clear, but also with a tail adapted for swimming, with vertebrae whose shape suggests the flexibility needed to drive the animal through water by vertical undulation. Its front limbs had wrists and elbows that could flex in ways a modern whale's cannot. Its body was webbed, or at least its feet were. It was, in short, a creature genuinely at home in both worlds, and what it did with that dual capability was something no modern whale could attempt. It went to sea, stayed at sea long enough to cross an ocean, and came ashore on the other side. The temptation when describing something like this is to frame it as a deliberate migration, as if Peregocetus's ancestors looked out across the Atlantic from Africa and decided to colonize the Americas. Nothing like that happened. What happened is that populations of protocetids living along the West African coast occasionally got swept offshore by currents, or followed prey out into open water, or for whatever combination of reasons found themselves in places where they had to keep swimming or die. Most of those individuals died. The few that happened to be carried by the right currents at the right time of year under conditions favorable to survival eventually made landfall somewhere on the other side. Some of those survivors encountered mates. Some of those mates produced offspring. And across many generations of this happening, with the vast majority of every transoceanic dispersal ending in failure, enough animals made the crossing to establish a breeding population in South America. The colonization wasn't planned. It was the statistical residue of a very large number of unsuccessful attempts. This matters because it is the pattern by which most major biogeographic dispersals actually happen. Islands get colonized by the lucky fraction of would-be colonists who don't drown. Continents get populated by lineages that happen to have members in the right place when the climate permits crossings. The evolutionary story of how life got distributed across the planet is, in most cases, a story about failure, about the enormous number of organisms who tried to cross water or ice or desert or mountain range and did not make it, with the successful few producing descendants who are, by definition, the only ones whose stories we can tell. Peregocetus is the record of one success. The failures left no fossils because most of them ended up in places where nothing fossilizes. They sank in deep water. They died at sea. They were eaten. The ocean does not preserve its dead the way coastal wetlands do. The fossil record for events like this is overwhelmingly biased towards survivors. Somewhere embedded in your own genome is the record of a different kind of survival. The lineage that produced you has crossed water too, though not in the same way. When modern humans dispersed out of Africa roughly 70,000 years ago and eventually reached Australia, Polynesia, and the Americas, the crossings involved water barriers that had to be bridged, sometimes by walking across glacial land bridges, sometimes by raft or canoe, sometimes, in the case of the remote Pacific Islands, by deliberate open ocean voyaging in vessels that must have tested the limits of the technology. Every person alive today is descended from some ancestor who survived one of those crossings. The ancestors who didn't survive left no descendants. The bias toward survivors that preserves Peregocetus's story is the same bias that preserves yours. You exist because your lineage is crossing at every generation from the first self-replicating molecule forward did not fail. If it had failed even once, you wouldn't be here to consider it. This is less a remarkable fact than a kind of tautology, but tautologies can be clarifying. The only ancestors you have are the ones who made it. What Peregocetus's body tells us, beyond the fact of the crossing, is something about the evolutionary timing of the transition. This animal still had legs. Good legs. Legs that worked on land. It could walk on a beach, climb out of the water, probably drag itself up the shore to rest in the sun. And yet it was already, functionally, a marine animal. Its tail was doing most of its locomotor work in the water, its feeding was almost certainly aquatic, and its dispersal pattern implies it was comfortable enough in open water to survive an oceanic crossing. This is the condition of an animal that has completed most of the behavioral and ecological transition to marine life without having completed the anatomical transition. The legs hadn't yet started to disappear. The pelvis was still articulated with the spine. The body still remembered how to walk. But the life being lived had essentially nothing to do with walking anymore. Evolution doesn't usually reorganize bodies in a single synchronized transition. It moves one system at a time, often with long lags between when a capability becomes behaviorally central and when the anatomy catches up. The whale lineage spent something like 5 or 6 million years in the Peregocetus-like condition, behaviorally marine, anatomically still quadrupedal, before selection began pulling the hind limbs aggressively toward reduction. During those millions of years, transoceanic dispersal was possible because the legs still worked on land when the animal needed them to. Once the legs started shrinking in earnest, the window for oceanic colonization by beaching-capable whales began to close. The lineages that descended from Peregocetus's Pacific population would, within another 10 million years, no longer be able to come ashore at all. By then, they wouldn't need to. The Ica Valley Desert today is the kind of place you can stand on and see nothing but stone and sky. When the wind is up, it moves small particles of sand across the flats with a faint hissing sound, and the bones of Peregocetus and its descendants emerge from the ground slowly, weathered out by erosion, waiting to be noticed by someone with enough time and enough reason to be looking. What was once the bottom of a warm tropical sea is now one of the driest places on the planet. Tectonics lifted it. The current cooled it. The rain stopped coming, and the whales that had lived in it 40 million years earlier became, eventually, a scatter of fossils in an empty country, findable only because so much of what once covered them has been stripped away. The lineage kept moving. In the Winterfield season of 2000, a paleontological team working in the Habib Rahi Formation of central Pakistan uncovered a small whale skeleton that, at first, seemed straightforward. Philip Gingerish, who was directing the excavation, saw teeth emerging from the matrix, and the teeth were small. His initial assumption was that the animal was a juvenile, perhaps an unusually well-preserved young protocetid, which would have been valuable, but not extraordinary. The team continued excavating carefully. More teeth came out, then part of the jaw, then ribs. And the ribs were the moment things stopped making sense, because the ribs were too large for the teeth. No juvenile has that proportion. A juvenile's ribs scale with its teeth. Both are small together, both grow together, and what Gingerick was looking at was an adult-sized rib cage holding juvenile-sized teeth. He kept clearing the matrix. By the end of that day, he understood what he had. The small teeth belonged to a fetus. The ribs belonged to the mother carrying it. This was, at the time of its discovery, the first fetal skeleton of an archaeocete ever found. The mother and her unborn calf had died together, something like 47.5 million years ago, and been buried in sediment fine enough to preserve the arrangement of both bodies in remarkable detail. 4 years later, in 2004, the team recovered a second skeleton from the same rocks, a fully complete adult of the same species, slightly larger, with somewhat larger canine teeth, which turned out to be an adult male. The species was formally described in 2009 in a paper in PLOS One, and Gingerich chose the name with some care. Maiacetus, from the Greek Maia, meaning mother, and cetus, meaning whale. The species name, Indohyus, came from a Roman fertility god. It was, unusually for paleontology, a name that referred explicitly to the evidence. The specimen that defined the species was a mother. What the Maiacetus pregnancy showed, beyond the simple fact that an early whale had died while carrying a near-term fetus, was the orientation of the calf inside the mother. This matters more than it might initially seem. In mammals, the way a calf is positioned before birth is not arbitrary. It is tightly tied to the environment in which birth takes place. Land mammals almost universally give birth head first because the head's emergence triggers the breathing reflex at the moment the newborn encounters air, and because head-first delivery is mechanically efficient for a body that will need to stand and move within minutes of its first breath. Marine mammals, modern whales, dolphins, seals, give birth tail first. The reason is straightforward. A whale calf born head-first underwater would take its first breath before it was free of the birth canal, and it would inhale water. Tail-first delivery keeps the calf's head inside the mother until the body is almost fully emerged, then releases it in a single final push. The mother and the assisting pod guide the newborn to the surface for its first breath within seconds. This is not a trivial adaptation. It is the difference between a viable birth and a drowned calf, and every fully marine mammal alive today has inherited some version of it. The Maiacetus fetus was positioned head first. What this means, if the positioning is reliable, and there has been some careful debate in the literature about whether fetal orientation in a single preserved specimen is genuinely diagnostic of birth location, a debate Thewissen in particular has raised, is that Maiacetus was still giving birth on land. 47.5 million years ago, after the lineage had been moving toward the water for at least 5 million years, after the ears had rebuilt themselves for underwater hearing, after the limbs had begun to shrink, after the body had committed to aquatic hunting and aquatic dispersal and aquatic everything else, the one thing that had not yet transitioned was birth. The animals still came ashore to produce their young. They still hauled themselves up onto beaches, found a sheltered spot, and delivered their calves the way a hoofed mammal delivers a calf, head first, on dry ground within reach of air. This is the kind of detail that quietly illustrates something important about how evolutionary transitions actually work. When a lineage starts to move into a new environment, different parts of the body and different parts of the life cycle transition at different rates. Some things change fast. The ear, as we've already seen, changed very early in the whale lineage because underwater hearing was the first strong selective pressure the ancestors of Pakicetus encountered. Locomotion changed more slowly because the physical demands of land and water are incompatible enough that a compromised body plan is workable for a long time. Feeding, sensory orientation, body shape, all of these reorganized across tens of millions of years at their own paces. Reproduction, in almost every transition we can track in the fossil record, is one of the last things to change. The machinery of birth is conservative. It involves so many interlocking anatomical and behavioral systems, pelvic geometry, uterine contractions, fetal positioning, maternal behavior after delivery, neonatal reflexes, that modifying any one of them tends to break the others. Evolution finds it much easier to reshape an adult body than to reshape how that body comes into existence. There is something worth sitting with here about what this implies for the animal itself. A pregnant Maiacetus, swimming through the shallows of the Tethys, hunting fish, foraging along the coastline, was living most of her life in water. Her ears told her where prey was. Her tail propelled her through the currents. Her limbs, though still functional, were more useful in the water than on land. And yet, somewhere in the last days or weeks of her pregnancy, she would have done what every one of her ancestors had done for hundreds of millions of years before the lineage ever approached the sea. She would have hauled herself onto a beach. She would have found a sheltered place above the tide line. She would have delivered her calf onto sand. and for a few weeks afterward she would likely have stayed there or nearby, nursing her young until the calf was strong enough to swim. The mental image of this is worth holding for a moment. A creature that in most respects no longer belongs on land, pulling its body up onto land anyway, because the older biology of being a mammal hasn't yet caught up with the newer biology of being a whale. Your own relationship to birth carries traces of the same lag. Human childbirth is notoriously difficult compared to that of most other primates because selection has given us an unusual combination of a large-headed infant and a bipedal pelvis that had to narrow to accommodate upright walking. The compromise our species made is suboptimal from an engineering standpoint. Human births take longer, are more dangerous to mother and child, and require more assistance than births in other great apes. We haven't solved it. We've arrived at a workable local minimum, a solution that works well enough most of the time at considerable cost, because no mutation in the last few million years has produced a cleaner fix. The pelvis we walk with and the pelvis we give birth through are the same pelvis, doing two incompatible jobs, and the resulting architecture is a compromise that neither a terrestrial quadruped nor a purely reproductive body plan would have arrived at. This is what suboptimal design looks like in a lineage caught between two regimes. Maiacetus, delivering her calf head-first on a Pakistani beach, was managing a similar compromise. A body mostly adapted for water, forced for reproductive reasons to return briefly to land, using an older terrestrial delivery orientation because the switch to tail first hadn't yet been selected for. Within another 5 or 6 million years, the lineage would work it out. The pelvis of Basilosaurids is no longer attached to the spine, which changes everything about the mechanics of birth, and by the time we get to fully modern whales, tail-first aquatic delivery is the norm. But getting there required restructuring the reproductive system piece by piece, and the Maiacetus skeleton is a snapshot of what the transition looks like when the machinery of birth has not yet been modified, when a whale, functionally, is still delivering its young the way a deer does. The reason the transition took so long is worth understanding in some detail because it illustrates a constraint that selection runs into repeatedly in cases like this. Reproduction is a tightly integrated system. The uterine shape that holds a head-first fetus is not the same uterine shape that holds a tail-first one. The pelvic architecture that accommodates head-first delivery has different load-bearing requirements than the architecture for tail-first. Maternal behavior after delivery, how the mother orients her body to present the newborn to air, how she assists the calf in reaching the surface, how she positions herself during nursing, involves muscle memory patterns and neural programs that evolved over millions of years of terrestrial mammalian history and cannot be rewritten overnight. Changing any one of these elements requires the others to change compatibly, or the mother dies, or the calf dies, and selection removes the mutation before it can spread. Evolution can only modify reproductive systems in small steps, each of which has to work on its own, and the number of small steps that preserve viable birth while moving toward a different delivery mode is small. It takes time, millions of years. The whale lineage took at least 5 million years to finish the transition, and probably longer. Something else Gingerich's team noticed in the 2009 paper was that the fetus had unusually well-developed teeth for a near-term mammalian calf. The first molars were already mineralized to a degree that suggested the newborn would have been able to feed on solid prey within days or weeks of birth, rather than relying exclusively on milk for an extended period. In zoological terms, this is called precociality, the condition of being born developmentally advanced, ready to move and eat and survive with minimal parental investment. Most marine mammals are precocial. Seal pups walk within minutes of birth. Dolphin calves swim with their mothers within hours. The precociality of the Maiacetus fetus suggests that this lineage had already committed developmentally to the pattern that modern marine mammals still follow. Calves born ready to participate in the lifestyle of their species almost immediately. The terrestrial delivery orientation was the last terrestrial feature to persist, embedded in a reproductive biology that was otherwise already marine. The preservation of the Maiacetus specimens is itself part of what makes them valuable. Fossils of pregnant animals are rare under any circumstances, and fossils of pregnant animals preserved with the fetus in a diagnostic orientation are rarer still. Most of what we know about the reproductive biology of extinct animals we know from inference, from the dimensions of the pelvic outlet, from the presumed body sizes of newborns estimated from growth patterns in juvenile skeletons, from trace fossils like tracks showing adults and small juveniles together. Direct fossil evidence of birth itself is almost unheard of. The Maiacetus pregnancy is one of the clearest windows we have into the reproductive biology of any extinct mammal lineage, and it tells us something we could not otherwise have known, that the whale lineage, very late in its transition to fully marine life, was still tied to land by the oldest constraint mammals carry, the constraint of how to bring a new body into the world. That constraint, across the next several million years, would finally yield. The lineage would stop coming ashore, the beaches would stop mattering, and the bodies would become something a land mammal would no longer recognize as related. Consider for a moment an animal that looks as if every detail of its body has been optimized for its way of life. It is 18 meters long, which is to say about the length of a city bus, with a serpentine body that moves through the water in long, powerful undulations. Its head is large and weaponized, with a jaw full of teeth shaped for different purposes at different positions, sharp conical teeth at the front for grasping prey, broader serrated teeth further back for shearing flesh from bone. Its forelimbs have been reduced to short flippers that steer rather than propel. Its tail, terminating in a horizontal fluke, provides the thrust. Its nostrils have migrated partway up the skull toward what will eventually become a blowhole in its descendants. Its inner ears are acoustically isolated from the rest of its skull, fully adapted for underwater hearing. It is the apex predator of the late Eocene oceans, unchallenged, dominant, a successful marine hunter of the kind that had not existed since the disappearance of the mosasaurs 30 million years earlier. And it has legs, not large legs, not functional legs in any meaningful sense. Near the rear of its body, about 2/3 of the way down a 16-meter skeleton, there is a small pelvis. The pelvis is not attached to the spine. It floats, suspended in muscle, serving no load-bearing role. Projecting from this pelvis are 2 hind limbs, each about 35 centimeters long. They have femurs, tibias, fused ankle bones, and 3 small digits. On an animal this size, they are roughly the length of a human forearm. They poke out from the body wall as 2 small appendages that cannot walk, cannot swim, cannot support any weight, and seem to the eye of anyone encountering the skeleton for the first time to be a kind of anatomical joke. A 16-meter serpent whale with the hind legs of a terrier, and the legs don't even reach the ground. The animal is Basilosaurus isis, and those legs are the single most important image in the entire story of whale evolution. The first Basilosaurus fossils were found in the early 19th century in the American South, and they were so strange that the paleontologist Richard Harlan, who described them in 1834, assumed they belonged to a giant marine reptile, a sea serpent in the old sense, some kind of massive Cretaceous holdover. He named the animal Basilosaurus, which means king lizard, and the name stuck even after the real identity of the creature was worked out. Within a few years, the anatomist Richard Owen had examined the specimens and pointed out that the teeth and skull were unmistakably mammalian, not reptilian. Basilosaurus was a whale. A weird one, but a whale. The name could not be changed because of the rules of zoological nomenclature, so to this day one of the most famous early cetaceans in the fossil record carries a name that describes something it is not. The truly remarkable Basilosaurus material came later, from Egypt. In the Western Desert, about 150 kilometers southwest of Cairo, there is a place called Wadi al-Hitan, the Valley of the Whales, where late Eocene marine sediments have been eroding for millions of years to expose the bones of an entire archaic whale ecosystem. Paleontologists working at Wadi al-Hitan have recovered more than 500 Archaeocete skeletons, mostly Basilosaurus isis and Dorodon atrox, many of them so complete that they preserve stomach contents, juveniles, pathologies, bite marks, and in 2015 a specimen that included the remains of its last meal, a Dorodon, still partially articulated in the gut cavity. Wadi al-Hitan is, for anyone interested in whale origins, one of the most important fossil sites on Earth. The UN declared it a World Heritage Site in 2005. Standing there, you can walk across ground that was once ocean floor and see, over the course of an hour, a dozen complete whale skeletons weathering out of sandstone under the desert sun. The hind limbs of Basilosaurus became clear only after the Egyptian material began to be recovered systematically in the 1980s, under the direction of Philip Gingerich and Elwyn Simons. In 1990, Gingerich, along with his colleagues B. Holly Smith and Elwyn Simons, published a paper in Science describing the hind limbs in detail. The legs had been known from fragmentary material for decades, but the Egyptian specimens allowed a complete reconstruction, and what the reconstruction showed was unambiguous. These were real legs, fully formed, with all the major skeletal elements a terrestrial mammal's leg has. Femur, tibia, fibula, tarsals, metatarsals, phalanges. Everything was there. Everything was small. The legs attached to a pelvis that no longer articulated with the sacrum, meaning in practical terms that no force generated by the legs could be transmitted to the spine. Whatever these legs did, they were doing it without any biomechanical connection to the rest of the skeleton. The paper's authors proposed that the most likely function of these vestigial legs was as copulatory guides, structures that helped align the male and female during mating, a plausible role given the geometry of Archaeocete bodies and the mechanical difficulty of underwater copulation in an animal this large. This interpretation remains the leading hypothesis, though it is not the only possibility, and it is worth being honest that the evidence is circumstantial. What we know for certain is that the legs could not support the animal's weight, could not propel it through water in any meaningful way, and could not have been used for any form of locomotion. What we don't know for certain is why selection didn't simply eliminate them over the intervening millions of years. This is the single most important question the Basilosaurus skeleton forces on anyone who takes it seriously, because it undermines a lazy but common assumption about how evolution works. The assumption goes something like this. If a structure has no function, it should disappear, because selection should eliminate anything that costs the organism energy to build and maintain without returning a survival or reproductive benefit. This assumption is wrong in the specific way that matters here. Selection does not eliminate structures just because they're useless. It eliminates structures only when they're actively harmful, when the cost of maintaining them exceeds the cost of removing them. Most vestigial structures, most of the time, are not expensive enough to be worth removing. They drift. They get smaller over millions of years because mutations that reduce them don't harm the animal, and random genetic drift accumulates in the direction of reduction. But the reduction is slow, and in the interim, the animal carries its residue. Basilosaurus had carried its legs for millions of years of aquatic life. The lineage had been fully marine since at least the time of Dorudon's ancestors, 30-something million years ago at that point in the Eocene. There had been plenty of generations for the legs to disappear entirely. But they hadn't. The legs were still there, still being built, still being grown in every embryo, still emerging from the body wall as small and functionally irrelevant appendages. The genetic machinery that produced them was still intact. The developmental program that placed them in the correct position on the body was still running. Selection had not removed them because selection had no strong reason to, and so they persisted as residues, as the fossil record of a body plan that the animal no longer needed. What makes the Basilosaurus legs particularly valuable for thinking about evolution is that they force the question of what we mean when we say a structure is adapted. A creationist argument, sometimes made, runs like this. If the legs are useless, why are they there? If evolution produces only adaptations, then the legs must have some purpose we haven't discovered. The answer is that evolution does not produce only adaptations. Evolution produces whatever selection permits to exist, and selection permits all sorts of things to exist that have no current function. It permits vestiges, it permits drift, it permits the residue of previous adaptations that have been overtaken by new selective regimes. The Basilosaurus leg is not an adaptation to anything in Basilosaurus's life. It is the half-erased signature of an adaptation that its ancestors had tens of millions of years earlier to a life that none of its descendants would ever lead again. Look at your own body. You have a small bone at the base of your spine called the coccyx. 3 to 5 fused vertebrae sitting where, for hundreds of millions of years of your lineage's ancestry, there was a tail. The tail was gone by the time your monkey ancestors were transitioning to apes. It has been gone in your lineage for something like 20 million years. The coccyx, meanwhile, is still there. Selection has never removed it, because removing it would involve a non-trivial reorganization of the pelvic floor musculature and provide essentially no benefit. It has been reduced from the functional tail of early mammals, through the stub tails of apes, down to the small residue in your own pelvis, but the reduction has not gone to completion. Your coccyx is, in miniature, what Basilosaurus's legs were. A piece of anatomical infrastructure that served a function in a distant ancestor was gradually reduced as that function became irrelevant, and has persisted in vestigial form because the cost of keeping it is small and the cost of removing it is not obviously smaller. The same principle applies across your body at multiple scales. Your appendix, the muscles in your outer ear that some people can still wiggle, residues of a time when mammalian ears rotated towards sound sources. The plica semilunaris at the inner corner of your eye, the pink triangle of tissue that is what remains of the nictitating membrane, the third eyelid still functional in reptiles and birds. The hair follicles on your arms that rise and trigger goosebumps when you're cold, firing a muscle that used to lift fur your ancestors no longer have. You are, in your own anatomy, walking through a museum of structures that no longer do what they were originally selected for, kept around because selection has not found a reason to remove them, and developmental constraints would make the removal non-trivial if it tried. This is what Basilosaurus's legs make visible at a scale large enough to be impossible to ignore. 16 meters of marine predator with 35 centimeters of useless terrestrial inheritance attached to it, because evolution is not a tidy process and does not clean up after itself. The elegance of the animal is real. The serpent body, the powerful tail, the predatory jaw, the acoustically isolated inner ear, all of these are genuinely adapted, genuinely shaped by selection for the life the animal actually lived. But the legs are the thing that tell you what evolution really is, because they are the things that don't fit. They are the signature of a lineage that changed so much its anatomy hadn't caught up, and the anatomy never would, and the legs would persist in reduced form in every Basilosaurus that ever lived until the lineage itself went extinct and the legs disappeared not by selection but by the elimination of the entire genus. They had another 30 million years in them, those legs, across the descendants of Basilosaurus that would produce all the modern whales. The embryonic hind limb buds that still form in every dolphin fetus, that still grow briefly and then get reabsorbed before birth, are the direct genetic descendants of the Basilosaurus leg. The same developmental program, the same Hox gene expressions, the same underlying architecture, now truncated so severely that the leg is born already gone, but still initiated every generation, in every dolphin and every orca and every blue whale, because the instruction set is still there, and the cell signaling cascade that builds a hind limb still starts and then stops before it gets very far. Evolution does not forget, it only quiets. Find the base of your spine. You can do this lying down without moving much. Just the fingertips of one hand reaching behind your lower back, tracing down along the vertebral column until you feel the small ridge of bone where your tailbone begins. You can feel it most clearly if you press gently into the muscle just above where the body curves inward at the sacrum. It is a small, fused structure, 3 to 5 vertebrae depending on the individual, pointing downward and slightly forward, embedded in the muscular and ligamentous tissue of your pelvic floor. It has a name, the coccyx, and it does almost nothing. It does a little. The muscles of the pelvic floor attach to it, and in that sense it plays a minor role in supporting the organs of the lower abdomen. But compared to what it once was, it is nearly nothing. Your coccyx is the final reduced residue of a tail, not a metaphorical tail, an actual vertebrate tail, the kind your mammalian ancestors carried fully functional for 200 million years before the lineage that produced primates began reducing it. Tree-dwelling early primates still had long tails for balance. Monkeys still have them. Apes lost them about 20 million years ago during a reorganization of the pelvic and lumbar region that had to do with body plan shifts toward brachiation and eventually, in some lineages, bipedal walking. By the time your direct ancestors arrived at something recognizably ape-like, the external tail was gone. The internal structure that once supported it, the articulated series of caudal vertebrae, got smaller and smaller until eventually the individual bones fused into a single small terminal segment, and even that segment does not emerge from your body. It is buried in flesh, invisible from the outside, a piece of your skeleton you can feel but not see. You carry it every day of your life. It does not trouble you. You probably go years without thinking about it. And it is, in its quiet way, the exact same kind of thing as the 35-centimeter legs of Basilosaurus. An anatomical residue of a life your species no longer leads, persisting because selection found no reason to finish removing it. The point of taking you through this exercise is not to teach you something new about your anatomy. The coccyx is not a secret. Anyone with an elementary biology textbook knows what it is. The point is to notice that the strangeness you probably felt in the previous chapter, looking at the image of a huge marine predator with tiny, useless legs, that strangeness is available to you in your own body right now, if you're willing to pay attention to it. Your body is not tidy. Your body is not optimized. Your body carries, embedded in its structure, the half-erased signatures of everything your lineage used to be and no longer is. The Basilosaurus legs and your coccyx are the same kind of thing. They differ in scale and visibility, but they are identical in what they tell us about the process that produced them. Let me slow down here, because this is the middle of the documentary, and it seems worth stopping for a few minutes to sit with what the story we've been telling actually means. Across the first 8 chapters, we've moved through about 60 million years of evolutionary history. We started in an empty post-impact ocean, followed the lineage of small Kashmiri artiodactyls that would eventually produce whales, watched Pakicetus begin rebuilding its ear for underwater hearing, seen Ambulocetus occupy the crocodile niche in shallow Pakistani deltas, traced Rodhocetus's ankle bones through a decades-long intellectual debate in paleontology, watched Peregocetus cross an ocean, observed Maiacetus deliver her calf on land, and arrived at Basilosaurus, the serpent whale apex, ruling the late Eocene seas, still carrying its useless legs. The story has the shape of a progression, and that shape is real in one limited sense. The fossils, arranged in the order they appear in the geological record, do show a cumulative increase in marine adaptation. Each successive stage is more aquatic than the one before it. The direction, in that specific sense, is real, but the direction is not what the story is about. Or rather, the direction is what the story looks like from the outside, from the retrospective vantage of someone watching the lineage's trajectory across the completed arc of 60 million years. From the inside, at any given moment in the actual historical process, there was no direction. There was only a population of animals, each individual doing what it could to survive and reproduce, with no awareness of what its lineage was becoming and no mechanism for caring. Pakicetus was not trying to become a whale. It was not aware of whales. Whales, as a recognizable body plan, did not yet exist. Pakicetus was a predator at a water's edge, making its living, mating when it could, dying when it had to, and producing offspring who happened to inherit whatever variations it happened to carry. Some of those offspring, in turn, happened to be born into generations where the populations carrying certain variants, slightly denser bones, slightly differently shaped inner ears, slightly more water-tolerant behavior, reproduced at marginally higher rates than their cousins. Across enough generations, those marginal advantages compounded. The population's average morphology shifted. New generations inherited the shifted baseline and were selected on it. That is the entire mechanism. There is no more to it than that. No foresight, no aim, no goal, no preferred outcome, no intelligent agent guiding the process, no evolutionary destiny pulling the lineage forward. Just populations of organisms in every generation, with some individuals reproducing more than others, and the balance of who reproduces more shifting slightly over time in response to the ecological conditions those individuals happen to encounter. The cumulative effect of this process, across tens of millions of years, can produce things that look extraordinarily designed. A Basilosaurus is, from a certain angle, an engineering marvel. Its hydrodynamics, its feeding apparatus, its sensory architecture, its integration of mammalian physiology into a marine lifestyle, all of these are remarkable. A human engineer presented with the task of designing an 18-meter marine predator for late Eocene conditions would have a hard time improving on what selection produced. The design quality is real, and nothing designed it. This is the thing that evolutionary biology is always, at its heart, trying to communicate, and it is the thing that the human mind finds most difficult to fully accept. We are pattern recognition machines that evolved in environments where seeing agents behind complex outcomes was usually a good survival strategy. When our hominid ancestors saw a rustling in the grass, it was safer to infer a leopard than to infer wind. The cost of being wrong about the leopard was death, and the cost of being wrong about the wind was only a minor false alarm. This cognitive bias, the tendency to detect agency and intention in the world around us, is deeply woven into how we perceive complex systems. When we look at a Basilosaurus, everything in our evolved cognition wants to ask, who made this? What was it designed for? What was the plan? The answer, nobody, nothing, there was no plan, is difficult to hold on to for long stretches of time. It slips. We reach for teleological language. We say the lineage evolved in order to do something, or for the purpose of accomplishing something, or with the goal of arriving somewhere. We say evolution wanted, or chose, or needed. All of these formulations import agency into a process that has none, and we do it not because we're being careless, but because the grammar of our thinking, shaped by the same evolutionary forces we're trying to describe, won't easily permit the accurate description. The accurate description is this. Imagine a very large number of dice, trillions of them, rolling continuously for many millions of years. Each roll represents a single organism's lifetime. Some rolls produce offspring, some don't. The rolls that produce offspring pass on a slightly modified version of the dice to the next generation. Across enough rolls, patterns emerge in the set of dice that end up in existence. Not because any individual die is aiming at the patterns, but because rolls that produce those patterns under the conditions of the environment tend to produce more subsequent rolls than rolls that don't. The patterns, in retrospect, look designed. They are not. They are what happens when you let the dice roll for long enough in an environment that rewards certain combinations more than others. The Basilosaurus legs are useful because they are the moment in the dice rolling process where the absence of design becomes difficult to ignore. If the lineage had been guided by any kind of intelligent engineering, the legs would have been removed the moment they became useless. They weren't removed. They persisted. They persisted because the process has no engineer in it, because selection cannot subtract what is not harming the organism, because evolutionary transitions always leave residue, because the dice that produced Basilosaurus carried, embedded in their patterns, the traces of every previous pattern that had ever existed in the lineage. Look at your own body again. Every one of the vestigial structures I mentioned in the previous chapter, your coccyx, your appendix, the small muscles around your outer ear that you probably cannot voluntarily move, the plica semilunaris at the corner of your eye, the goosebumps on your skin that rise from hair follicles lifting a coat you no longer have, is a piece of evidence that the process that produced you does not tidy up. You are not a designed animal. You are a statistical residue, a pattern that emerged across hundreds of millions of years of rolling dice carrying the half-erased signatures of everything that was in your lineage before you. Some of those signatures are 200 million years old. Some are 40 million years old. Some are recent enough to have happened within the last few million years of hominid evolution. You are not an organism that was made. You are an organism that was accumulated. And that is not a diminishment of what you are. That is what the honest description actually looks like. The accumulation is immense. The process that produced your body ran for 3.8 billion years, and every ancestor in that line survived long enough to reproduce without a single exception. Because if any one of them had failed, you would not exist. The unbroken continuity of the thread from the first self-replicating molecule to the mind hearing this sentence is, at minimum, as remarkable as any designed thing could possibly be, and probably more so because designed things can be made quickly and for specific reasons, whereas this thing was produced slowly by no one for no reason, and yet it produced you. A Basilosaurus, cruising the late Eocene oceans with its useless legs poking out from its flanks, was at the end of exactly the same kind of thread. Its ancestors had each, in turn, survived long enough to reproduce, stretching back through Pakicetus, through the common ancestor with artiodactyls, through the common ancestor with all mammals, through the common ancestor with all tetrapods, through the first lobe-finned fish, through the first vertebrates, through the first multicellular eukaryotes, through the first eukaryotic cells, back to the origin of life itself. Every Basilosaurus that ever lived was the current terminus of the same continuous thread of reproduction that, in a different branch, would eventually produce the human hearing this description. Its useless legs were as much a part of that thread as your coccyx is part of yours. The thread continues. It has never at any point broken. The animals you share it with include whales, and they always will. Around 34 million years ago, the Earth's climate changed rapidly enough that paleoceanographers have given the event a specific name. They call it the Eocene-Oligocene transition, or sometimes the EOT for short, and what happened in that relatively narrow geological interval, a few 100,000 years, perhaps less, is that the planet moved from the long, hot, greenhouse conditions that had characterized the Eocene into a much cooler regime that would persist with fluctuations through the Oligocene and into the subsequent geological epochs. The transition was driven by several interlocking factors, most significantly the opening of ocean passages around Antarctica as the southern supercontinent finished breaking up. When the Drake Passage opened between South America and the Antarctic Peninsula, and when the seaway between Antarctica and Australia widened, a new ocean current could form, a circumpolar current that circled Antarctica without interruption, thermally isolating the continent from the warmer waters further north. Within a geologically short period, ice began to accumulate on Antarctica for the first time in tens of millions of years. Sea levels dropped as water was locked up in the growing ice sheets. Deep water circulation patterns reorganized across every ocean. Temperatures at the surface of the sea fell by several degrees on average, with much larger drops in polar waters. For the marine animals that had adapted to the warm Eocene oceans, this was a crisis. The Archaeocete whales, the Basilosaurids and their relatives, had spent the previous several million years as the dominant large predators of tropical and subtropical shallow seas, feeding in ecosystems structured around the warm water productivity of the late Eocene. That productivity reorganized as the climate cooled. The distribution of prey species shifted. Coastal habitats that had been teeming with fish and small marine mammals became, in some regions, productive in entirely new ways, while other regions collapsed. The ecological framework that had supported Basilosaurus and Dorudon for millions of years began to come apart, and the Basilosaurids, whose bodies had been optimized for a specific set of warm water conditions, found themselves poorly suited to the new world that was emerging. Most of them went extinct. This is worth being precise about because the extinction was not total. A few basilosaurid lineages survived the transition, at least for a while, and the genus Basilosaurus itself persisted into the early Oligocene before disappearing. But the bulk of the Archaeocete diversity that had characterized the late Eocene did not make it across the boundary. Of the dozen or more protocetid and basilosaurid genera that had been diversifying across the world's oceans in the Middle and Late Eocene, only a handful left descendants in the Oligocene. The Ambulocetids were already long gone. The Remingtonocetids had vanished millions of years earlier. The Protocetids had mostly disappeared before the EOT, replaced by the more fully aquatic Basilosaurids. And now the Basilosaurids themselves, the apex predators of their era, were being culled by the climate. The lineage that would eventually produce every modern whale, every blue whale, every sperm whale, every dolphin, came through this bottleneck not as a diverse, flourishing group, but as one or two surviving branches scraping through a narrow geological window while most of their cousins died. The story of almost every major evolutionary radiation includes a moment like this. A lineage diversifies, succeeds, dominates its niche for some period of time, and then encounters an environmental change it is not ready for. Most of the lineage dies, a small fraction survives, usually by having happened to possess traits that were neutral or even mildly disadvantageous during the period of dominance, but become useful in the new conditions. The survivors radiate into the ecological vacancies left by the dead, and the next round of diversification begins. We are, in the standard evolutionary account, usually at least one extinction bottleneck downstream from whatever lineage we're interested in studying. The mammals we know well are the descendants of the lineages that survived the end Cretaceous extinction. The tetrapods as a whole are descended from the lineages that survived the end Permian extinction. And the modern whales are descended from the basilosaurid lineages that survived the Eocene-Oligocene cooling. What came out the other side of this bottleneck was a radically reorganized cetacean fauna. The basilosaurid body plan, long serpentine surface cruising, was not what the post-EOT oceans selected for. The new conditions favored animals with different capabilities, animals that could hunt in colder water, animals that could dive more deeply, animals that could exploit the newly productive polar and subpolar waters where ice-driven mixing was generating massive seasonal blooms of plankton and fish, and animals that could do one of two things very well: either filter feed on the small prey that was suddenly available in enormous quantities, or hunt single large prey in environments where vision was increasingly limited by depth or by turbidity or by darkness. Two groups of descendants emerging from the basilosaurid bottleneck happened to be well positioned for these two different opportunities. One group would become the Mysticeti, the baleen whales, specializing in bulk filter feeding on concentrated prey. The other would become the Odontoceti, the toothed whales, specializing in active pursuit of individual prey, increasingly using sound rather than sight to locate it. These two lineages diverged from their common basilosaurid ancestor sometime in the early Oligocene, perhaps around 35 million years ago, and within a few million years of the split, they had become recognizably distinct. The split is the most important branching event in whale evolutionary history after the original land-to-sea transition itself, because it set the stage for everything that modern whale diversity represents. What's worth noticing about this split is that it wasn't driven by some grand evolutionary logic. Nothing decided that cetaceans should specialize into filter feeders and pursuit predators. What happened was that the post-EOT oceans contained two different ecological opportunities that rewarded two different sets of traits, and the populations of basilosaurid descendants happened to have enough genetic variation among them that some individuals drifted in one direction while others drifted in the other. Over enough generations, the drifting populations became reproductively isolated. Once reproductive isolation was in place, the two lineages could no longer exchange genes, and each could specialize further without being pulled back toward the ancestral middle by interbreeding. The specialization deepened, the lineages diverged, and what started as two populations of generalists within a single species became, across 10 or 15 million years, two entirely different kinds of whale. This is how almost all major evolutionary radiations actually work, not through some kind of adaptive leap or evolutionary breakthrough, but through the slow accumulation of small genetic differences between populations that have stopped exchanging genes. Once gene flow stops, the populations drift apart. Selection pushes them further apart whenever their environments differ. Random genetic drift pushes them apart even when selection is neutral. Across enough time, they become different species, then different genera, then different families, then eventually different suborders of cetacean, each with its own distinctive body plan and its own distinctive way of making a living. Your own species is the product of exactly this kind of process, played out at smaller scales and over shorter time spans. The lineage that became humans diverged from the lineage that became chimpanzees something like 6 or 7 million years ago, and the divergence started as two populations of what was probably a single ape species finding themselves in slightly different environments and drifting apart genetically. Everything that has happened since, every morphological change, every behavioral innovation, every cognitive development that distinguishes humans from chimpanzees, began from that initial separation. And once the separation was in place, the accumulation of differences was essentially unstoppable because there was no longer a mechanism for bringing the populations back together. The whales did the same thing twice at the Eocene-Oligocene transition. Mysticetes and Odontocetes began as one population. The climate shifted, the opportunities divided, the populations drifted apart. And within a few tens of millions of years, they had become the two distinct cetacean lineages that now, collectively, constitute every whale alive. There's a detail about the post-EOT whale fauna that deserves special attention because it points to one of the most elegant byproducts of the whole transition. The basilosaurid lineage that made it through the cooling carried with it, from its archaic ancestry, a particular set of physiological adaptations, a myoglobin-rich musculature, a circulatory system already capable of supporting extended breath holds, an inner ear already acoustically isolated from the skull. None of these had evolved specifically for the conditions of the Oligocene oceans. They had evolved in the Eocene, for Eocene reasons, in animals that would eventually die out. But they happened to be exactly what the descendants of those animals needed to exploit the new conditions. The basilosaurids passed on a toolkit that their own lineages couldn't use anymore, but which turned out to be exactly the right starting point for what came next. The Mysticetes would take that toolkit and refine it for filter feeding in cold, upwelling zones. The Odontocetes would take it and refine it for echolocation and deep diving pursuit. Neither group had to build their core adaptations from scratch. They inherited most of what they needed, and selection only had to make adjustments. The redness of whale blood, incidentally, was already there too. It had always been there. The iron in the hemoglobin that carries oxygen through your own bloodstream, binding and releasing it at each pass through the lungs and the tissues, is the same iron, carried in a very similar hemoglobin protein that carries oxygen through every whale. The red color is a consequence of the chemistry of iron and oxygen, the way iron-oxygen complexes absorb some wavelengths of light and reflect others. Nobody selected for redness. It is a chemical byproduct of the way the molecule works, present across every vertebrate that uses iron-based oxygen transport, and it would look exactly the same in a basilosaurid bleeding out into Eocene water as it would in a modern blue whale bleeding into the Antarctic. The color is not an adaptation. The color is what you get physically when you build an oxygen transport system out of iron and hemoglobin. And every whale and every human carries the same color for the same reason. The lineages that came through the Eocene-Oligocene bottleneck carried this inheritance forward. They would take the next 30 million years to become something the Eocene oceans could never have produced. There is a misconception about cetacean taxonomy that is surprisingly widespread, even among people who consider themselves reasonably well informed about animals, and it is worth naming directly because correcting it helps organize everything that follows. The misconception is that whales and dolphins are two different kinds of animal, that whales are the large marine mammals and dolphins are a separate, smaller category with some kind of fundamental taxonomic distinction between them. This is not how the biology works. Dolphins are whales. Every dolphin species that swims in the oceans today is a cetacean, and more specifically, an odontocete, a toothed whale. A bottlenose dolphin is as much a whale as a sperm whale is. The word dolphin is a common language label applied to certain smaller members of the odontocete suborder, and it does not correspond to any natural taxonomic boundary. If you were to construct a phylogeny of living cetaceans without knowing any of the common names, you would find no place to draw a line separating whales from dolphins. The line does not exist in nature. It exists only in English vocabulary. This matters because the real taxonomic division among modern cetaceans is elsewhere, and it is the division we began tracing at the end of the previous chapter. Every living whale belongs to one of two suborders. The Mysticeti, the baleen whales, includes the blue whale, the humpback, the fin whale, the grey whale, the right whales, the bowhead, and the minke, among others. About 15 species in total, all of them filter feeders, all of them large, all of them equipped with baleen plates rather than teeth in the adult mouth. The Odontoceti, the toothed whales, includes everything else cetacean that swims in the oceans today. Sperm whales, beaked whales, orcas, which are technically the largest dolphins, pilot whales, porpoises, river dolphins, belugas, narwhals, and all the species most people think of as dolphins. About 76 species in all, every one of them equipped with teeth and, in almost every case, with a functioning echolocation system. These two groups share a common ancestor that lived in the early Oligocene, about 35 million years ago, the basilosaurid descendant that survived the Eocene-Oligocene bottleneck we traced in the last chapter. From that single ancestor, the two lineages diverged, specialized, and over the following tens of millions of years became as different from each other as two sister groups of cetaceans could become. The depth of the divergence between Mysticeti and Odontoceti, measured in genetic terms, is considerably greater than the divergence between humans and any other living primate. Mysticetes and Odontocetes have been separated for roughly 5 times as long as humans have been separated from chimpanzees. They are deeply, fundamentally different animals. They just happen to both be whales. What this divergence produced was two entirely different solutions to the problem of being a large marine mammal. Both solutions involve size, because size remains advantageous for thermoregulation in cold water and for metabolic efficiency during long-distance travel. Both solutions involve breath-hold diving, because both lineages inherited the basilosaurid physiology that supported it. Both solutions involve sophisticated underwater hearing, because both inherited the acoustically isolated inner ear that had been present since Pakicetus. But beyond these shared starting points, the two lineages moved in opposite ecological directions, and the divergence shaped everything about their subsequent evolution. The Mysticete solution is to eat small things in enormous quantities. A blue whale feeding in the Antarctic takes in up to 4 tons of krill per day during the feeding season, which it accomplishes by lunging into dense patches of the tiny shrimp-like crustaceans with its mouth open, engulfing several tons of prey-laden water in a single gulp, and then filtering the water out through its baleen plates while retaining the krill inside the mouth. The baleen itself is a keratinous structure, the same kind of tissue as your fingernails and hair, that grows in sheets from the upper palate and hangs down in curtains that strain small prey from large volumes of water. This feeding strategy works only at scale. A small filter feeder can find enough food to survive. A truly large filter feeder can find enough food to thrive, because the surface area to volume ratio of the mouth and the volume of water that can be processed per unit time scale favorably with body size. The blue whale is the largest animal that has ever lived, and the reason it is the largest animal that has ever lived is specifically because filter feeding on dense prey patches rewards size in a way that almost no other feeding strategy does. The Odontocete solution is the opposite. Rather than processing large volumes of water for small prey, Odontocetes hunt individual prey items using active location, usually echolocation, sometimes vision in clearer water, sometimes a combination. A sperm whale diving to 2,000 meters in pursuit of a single giant squid is taking the Odontocete strategy to its extreme. The animal has located the squid through sound, usually its own echolocation clicks, and is pursuing it as a single hunter going after a single meal. This kind of hunting rewards different traits than filter feeding does. It rewards sensory sophistication, the ability to construct an accurate three-dimensional picture of the environment from acoustic returns. It rewards cognitive flexibility, the ability to track prey movements, predict escape patterns, coordinate with other hunters in some species. It rewards precision rather than bulk. And it permits, though it does not require, much smaller body sizes than filter feeding does, because a 6-foot dolphin can make a living pursuing individual fish in a way that a 6-foot filter feeder could not. The result is that Odontocetes span an enormous range of body sizes, from the 6-foot vaquita porpoise up to the 60-foot sperm whale, while Mysticetes are almost all large. The smallest living mysticete, the pygmy right whale, is still over 6 meters long, and most mysticetes are much larger. Size for mysticetes is not optional. Size for Odontocetes is one option among many. This divergence is an example of what evolutionary biologists call convergent evolution, though the convergence here is not between the two cetacean lineages, but between each of them and unrelated marine predators that have occupied similar niches. The mysticete body plan, large, streamlined, filter-feeding, exploiting concentrated planktonic prey, has been convergently arrived at by other marine animals multiple times. Whale sharks, the largest living fish, are filter feeders with a broadly similar ecological role. Basking sharks are similar. Megalodon, the extinct giant shark of the Pliocene and Pleistocene, was a pursuit predator rather than a filter feeder, but occupied the large marine apex niche that mysticetes now share. The filter-feeding giant is a recurring body plan in the oceans, appearing in unrelated lineages whenever sufficient quantities of concentrated small prey are available to sustain one. Mysticetes arrived at it from an ancestral lineage of toothed mammals. Whale sharks arrived at it from an ancestral lineage of cartilaginous fish. The two starting points could hardly be more different. The ending points are recognizably similar because the physics of being a giant filter feeder in marine environments constrains the solutions available. The odontocete body plan is convergent with the ichthyosaurs of the Mesozoic, the extinct marine reptiles that occupied broadly similar ecological roles as pursuit predators in Mesozoic oceans. The resemblance between a dolphin and a modern depiction of an ichthyosaur is, at first glance, almost eerie. Both have streamlined fusiform bodies, dorsal fins placed roughly 2/3 of the way down the body, horizontal tail flukes, though the ichthyosaurs had vertical tail flukes in a difference that betrays their reptilian rather than mammalian heritage, and elongated rostra filled with conical teeth suited to grasping small, fast-moving prey. No common ancestor of dolphins and ichthyosaurs had any of this body plan. The two lineages arrived at the same solution by different paths, separated by hundreds of millions of years because the pursuit predator niche in open marine water selects strongly for a particular combination of hydrodynamic features, and the number of workable combinations is small. This is the same convergence principle we saw in the ambush predator niche with Ambulocetus and the crocodilians. Same logic, different niche, different body plan. When ecological pressures are sustained and physically specific, the solutions that selection can produce are constrained. Lineages as distant as marine reptiles and marine mammals can arrive at essentially the same body plan if the pressures are the same. What looks like design from the outside is the signature of a constrained solution space, a handful of workable answers to a physical problem, arrived at independently by any lineage that happens to face the problem. Your own color vision is the relevant body archive moment here, because the cetacean lineages exemplify what happens when a sensory modality stops being selected for. Most mammals are dichromats. They have two types of color-sensitive cone cells in their retinas, and they see the world in something like a reduced two-channel version of what a full-color camera would capture. Many primates, including humans, are trichromats. We have 3 types of cones and see a richer color space than most mammals do. Trichromacy in primates is a relatively recent innovation. reacquired after most mammalian lineages had simplified their color vision during an earlier period of nocturnal evolution. In cetaceans, the simplification has gone further. Modern whales are monochromats. They have only one type of functional cone, or none at all in some species, and they see the world essentially in grayscale. The genes for the other cone types are still in their genomes, but they have been inactivated, pseudogenes, broken by mutations that accumulated once the selective pressure to maintain them disappeared. Whales don't need color vision because their world is mostly dim, mostly monochromatic already, and mostly navigated by sound rather than by sight. Selection stopped maintaining the color vision genes, and drift broke them. Your trichromacy, sitting on top of your ancestral mammalian dichromatic baseline, is the result of the opposite process. Primates reacquired a third cone type because primate ancestors happened to be eating fruit in a way where distinguishing red from green from yellow was nutritionally useful. The same gene family was selected for in our lineage and against in the whale lineage, and the difference is entirely about what environment each lineage happened to find itself in. Neither outcome is better. Neither is worse. Each is what selection produces when it is pushing in a particular direction for long enough. This is what the Mysticeti-Odontoceti split is at the deepest level. Not a grand evolutionary plan, not a designed bifurcation of cetacean possibility space, just two populations of the same basilosaurid descendants happening to find themselves in slightly different ecological circumstances, drifting apart genetically, diverging over tens of millions of years into two different ways of being a marine mammal. The divergence looks profound from the outside, from the vantage of an observer looking at a blue whale and a dolphin and noticing how different they are. From the inside, at any moment along the way, it was just populations of animals doing what their local conditions rewarded, accumulating small differences, eventually unable to interbreed, continuing on their separate trajectories. The depth of the current divergence is measured not in any individual step along the way, but in the sheer number of steps, each small, each locally unremarkable, compounding across something like 200 generations per million years for 35 million years, until the two lineages became unrecognizable to each other. One of them in particular was developing something that had never existed before. A sperm whale at 2 kilometers of depth is operating in an environment that human intuition is poorly equipped to imagine. Sunlight has not penetrated this far for billions of years. The water around the animal is at a temperature just above freezing, about 2 degrees Celsius. The pressure is approximately 200 times what it is at the surface, which would crush a human within seconds. There is no visual information available at this depth. Nothing glows, nothing reflects, nothing gives away its position by shape or shadow. The only things that move down here are the animals themselves, the slow drift of deep currents, and the fall of organic debris from the sunlit waters kilometers above. And yet, in the middle of this absolute darkness, a sperm whale is hunting. It has located, with a precision that would be impressive in full daylight, a giant squid at a distance of perhaps 50 meters. The squid is trying to escape, the whale is adjusting its approach, and none of this is happening through sight, all of it is happening through sound. The sperm whale is clicking. Short, sharp, high-intensity acoustic pulses generated by forcing air through specialized structures in its nasal passages called phonic lips, amplified and focused by the spermaceti organ, the enormous oil-filled structure that takes up most of the whale's massive head, and projected forward into the water as a narrow beam of sound. The clicks travel outward at about 1,500 meters per second, which is 4.5 times faster than sound in air. They hit the squid. They reflect. They return to the whale, arriving at the lower jaw, where a channel of fatty tissue conducts them back through acoustic windows into the inner ear. The whale's auditory system processes the returning echoes with something close to the speed of thought, extracting from them information about the squid's position, orientation, size, shape, texture, and direction of motion. The whale adjusts. It clicks again. The squid shifts. The whale tracks the shift. And within perhaps a few seconds, the hunter is in striking distance of prey it has never seen and never will. This is echolocation, and it is one of the strangest things any mammal does. Your own auditory system is a passive receiver. Sound arrives at your ears from the environment, and your brain interprets what arrives. The whale's system is active. It produces sound deliberately in order to reveal things about the environment that passive listening could not reveal. It is using sound the way you use light, projecting it outward, reading what comes back, constructing from the returns a three-dimensional model of the world. The closest human analogy is probably radar or sonar technology, which we invented in the 20th century. The sperm whale has been doing it for somewhere around 30 million years. The origin of Odontoceti echolocation is one of the most interesting evolutionary puzzles in the cetacean story, because it is both ancient and remarkably specific. Echolocation in modern toothed whales requires an integrated suite of adaptations: the specialized phonic lips for sound production, the spermaceti or melon organ for acoustic focusing, the asymmetrical skull that orients the sound beam, the fatty tissue channel in the lower jaw for echo reception, the cochlea tuned to the particular high frequencies the animal produces. All of these features have to be present together for the system to work. A whale with only some of them would have no functional echolocation. This raises an obvious question about how the system evolved, since natural selection cannot produce complex multi-component systems all at once. The components must have come together one at a time, and at least some of them must have been doing something useful before they were doing echolocation. What the fossil record shows with some precision is that echolocation evolved during a remarkably short geological interval in the early Oligocene. The earliest fossils that can be confidently identified as stem Odontocetes come from rocks dated to between 30 and 32 million years ago, primarily in the southeastern United States and in New Zealand. A group called the Xenorophidae, which includes animals like Echovenata sandersi, represent the earliest diverging odontocete lineages. And when paleontologists used CT scanning in the 2000s to examine the inner ears of these early odontocetes, work led by researchers including Travis Park, Rachel Rasikut, and Jonathan Geisler, they found something important. The cochlea of xenorophids were already specialized for ultrasonic hearing. The same high-frequency sensitivity that modern toothed whales use for processing echolocation returns was present in the very earliest Odontocetes, emerging essentially at the same geological moment that the lineage itself emerges. Echolocation, or something very close to it, appears to have been one of the defining features of Odontocetes from almost the beginning of the group. This is striking because of how fast it happened. The Eocene-Oligocene transition occurred about 34 million years ago. The first recognizable Odontocetes appear about 30 million years ago. Somewhere in the 4 million years between those dates, which is short by the standards of major evolutionary innovations, the full suite of echolocation adaptations was assembled from ancestral components. The speed of this assembly has been used by some creationist writers to argue that echolocation could not have evolved by natural selection. The time window is too short. The argument is wrong, but understanding why it is wrong is useful for understanding how evolution actually operates when it is producing novel capabilities quickly. The key point is that echolocation was not built from nothing in 4 million years. It was assembled from components that were mostly already present in the Basilosaurid ancestry. The acoustically isolated inner ear had existed since Pakicetus, 50 million years ago. The fat-filled lower jaw channel for sound reception was present in Basilosaurids, originally used for passive underwater hearing. The capacity to produce sounds with the upper respiratory system was ancient. All mammals can vocalize, and early whales had modified the vocal apparatus for underwater communication long before echolocation became an issue. The spermaceti organ, in particular, appears to have originated not as a sonar-focusing device, but as something involved in buoyancy control. The waxy oil inside it changes density with temperature, allowing the whale to adjust its vertical position in the water column by cooling or warming the organ. That was its original function, probably in early Odontocetes. Sonar focusing was a later exaptation of a structure that had already evolved for a different purpose. The 4 million years of rapid echolocation evolution, then, was not a matter of building all those components from scratch. It was a matter of repurposing and coordinating components that already existed, modifying their proportions, tuning the cochlea to higher frequencies, adjusting the geometry of the skull to direct the sound beam, and shaping the spermaceti organ so that its acoustic properties became more important than its buoyancy properties. None of these individual changes required a large evolutionary leap. Each one was a small modification of an existing structure. The full echolocation system emerged because a lineage of early Odontocetes happened to carry all the necessary precursors, and the selective pressure to hunt in low visibility conditions was strong enough for long enough that the precursors drifted together into an integrated functional system over tens of thousands of generations. This is what complex evolutionary innovations almost always look like when you examine them closely. They appear from a distance to require extraordinary leaps. They turn out, on closer inspection, to be assemblies of components, each of which was already doing something else, selected into new functional combinations by sustained pressure over geologically moderate periods of time. The eye evolved this way. Flight evolved this way, venom systems evolved this way, echolocation evolved this way. The illusion of a sudden miraculous innovation is created by the retrospective vantage, looking at a modern sperm whale and seeing the integrated echolocation system. It is easy to think of it as a single adaptation, when it is actually a tight coordination of many older adaptations that only recently started working together. Your own middle ear is the relevant body archive here, because your 3 ossicles, the malleus, incus, and stapes, are the same 3 bones the whale uses, modified for different purposes in the 2 lineages. In you, they transmit airborne sound vibrations from the eardrum to the inner ear, amplifying the signal so that it can excite the fluid-filled cochlea. In the whale, the same 3 bones are structurally reinforced, repositioned, and integrated into a system of fatty tissue channels and acoustic isolation air sinuses that together enable directional underwater hearing at frequencies far beyond what human ears can perceive. The evolutionary history of these 3 bones is one of the most extensively studied examples of exaptation in the mammalian record. They began as jaw bones in the reptilian ancestors of mammals about 200 million years ago. During the early evolution of mammals, they migrated into the middle ear and took on their auditory function. In the whale lineage, that auditory function was then further modified for underwater conditions, with the same 3 bones being repositioned and acoustically isolated to permit directional hearing in a medium where ordinary mammalian ears cannot localize sound. And then, in Odontocetes, the same system was further refined to process the specific high-frequency returns of active echolocation. Each of these steps used what the previous step had produced. None of them started from scratch. The 3 bones that transmit the sound of this sentence from your eardrum to your inner ear are the same 3 bones that allow a sperm whale in total darkness 2 kilometers below the surface of the Pacific to resolve the position and velocity of a squid by the acoustic returns of its own clicks. You hear me? The whale sees the squid. The underlying hardware is the same. There is one more thing about Odontoceti echolocation worth sitting with for a moment, because it connects to the convergence argument we have been developing across this documentary. Echolocation has evolved independently at least twice in mammals. Once in Odontocetes, sometime in the early Oligocene, and once in Microchiropteran bats at roughly the same geological moment during the Eocene-Oligocene transition. The two lineages share no common echolocating ancestor. The last common ancestor of whales and bats lived more than 90 million years ago and had no sonar capabilities at all. And yet, both lineages arrived at echolocation independently, within a narrow window of time, under ecological pressures that rewarded the ability to hunt in low visibility conditions, darkness for bats hunting insects at night, depth and turbidity for Odontocetes hunting in the deep or murky waters. When molecular biologists examined the genes that encode the proteins of the echolocation system in both lineages, they found something genuinely surprising. The same genes in both bats and whales had accumulated the same amino acid substitutions in similar patterns during their independent evolutions of echolocation. The molecular level changes required to produce high-frequency hearing from standard mammalian hearing are apparently limited enough that two unrelated lineages facing similar selective pressures arrived at essentially the same genetic solutions. This is sometimes called molecular convergence, and it is one of the more striking demonstrations that the space of workable evolutionary solutions, even at the level of individual protein modifications, is much smaller than one might naively expect. The sperm whale, clicking in the dark at 2 kilometers down, and the little brown bat, clicking in the dark at 2 meters above a North American pond, are using the same acoustic strategy, evolved independently, encoded in modified versions of the same genes, selected for by the same underlying logic of how sound and darkness interact. Nothing chose this convergence. It is what emerges when constraints are sustained and the solutions available are few. If you could look inside the mouth of a developing baleen whale fetus, say a blue whale or a humpback, about halfway through gestation, suspended in amniotic fluid inside its mother's uterus, you would see something the adult animal will never have. You would see teeth, small, conical, unmineralized teeth, arranged along the margins of the jaws in rows roughly where you would expect teeth to be in any mammalian mouth. They develop on the normal mammalian embryological schedule, initiated by the same tissue interactions that produce teeth in every mammal, following the same signaling cascades that generate the tooth buds in a human embryo at about 6 weeks after conception. They grow for a few weeks, and then before the fetus is born, they are reabsorbed. The tissue that produced them is dismantled and taken back into the body. By the time the calf emerges from the birth canal, tail first and breathing its first air, it has no teeth at all. It will have no teeth for the rest of its life. What it will have instead, growing from the upper palate where its teeth used to be, is baleen, long, keratinous plates arranged in closely spaced rows, fraying at their inner margins into hair-like filaments that will filter small prey from enormous volumes of seawater. Your own embryological development did something similar, though the structure that was initiated and then reabsorbed was different. At approximately 6 weeks of gestation, your embryo had a tail, a real one, with vertebrae that extended beyond what is now your coccyx, visible in ultrasound images of early-stage fetuses as a distinct posterior projection. Over the following weeks, the tail was gradually reabsorbed. The individual vertebrae fused or disappeared, the soft tissue that had extended beyond the body wall regressed, and by the time you were born, all that remained of the tail was the internal coccyx we talked about several chapters ago. You went through a phase of being a tailed animal and then stopped being one inside your mother weeks before you were ready to be born. The Maiacetus fetus goes through a phase of being a toothed animal and then stops being one inside its mother weeks before it is ready to be born. The parallel is exact. Same kind of developmental program, same kind of evolutionary residue, different specific structure. This is the evidence that modern baleen whales evolved from toothed ancestors, and it is some of the cleanest evidence any evolutionary transition has ever produced. The toothed ancestry is not a matter of inference from distant similarities, it is visible directly every generation in the embryos of every living mysticete. The genetic machinery for producing teeth is still intact. The developmental program that assembles them is still running. Selection has not removed the program, it has only shortened it, truncating the tooth production sequence before the teeth can mineralize and erupt. If you were to experimentally manipulate a mysticete embryo's development, extending the tooth production sequence or suppressing the reabsorption, you would presumably get a baleen whale born with teeth. Nobody has done this experiment for obvious ethical reasons, but the genetic and developmental evidence suggests it would work. The teeth are there. The genome still knows how to make them. Only the timing has been modified. The story of how teeth became baleen in the Maiacetus lineage was reconstructed in detail in the mid-2000s, primarily through the work of Thomas Desmarais at the San Diego Natural History Museum and his colleagues, building on earlier fossil discoveries and on molecular evidence from modern whale genomes. The reconstruction goes like this. The earliest mysticetes, the toothed ancestors, looked broadly similar to Basilosaurids and retained functional teeth that they used for predation on small fish and squid, much like modern dolphins do. At some point in the Oligocene, probably between 30 and 25 million years ago, a lineage of these toothed mysticetes began evolving nutrient foramina, small holes in the bones of the upper palate through which blood vessels passed to supply soft tissue on the palate surface. These foramina are diagnostic features that appear in the fossil record before baleen itself does, and Desmarais' team argued, convincingly, that they represent the vascular infrastructure required to grow baleen. In other words, the blood supply for baleen evolved before the baleen itself did. This makes sense. You cannot grow a keratinous filter-feeding structure on your palate unless you have the blood supply to nourish it. The infrastructure had to come first. What came next, in the fossil record, was a group of whales that had both functional teeth and the palatal foramina that imply the beginnings of baleen. Species like Eocene weltoni, known from Oligocene rocks in Oregon, show this mosaic condition. Eocene had teeth. It could catch prey by conventional predation, but it also had the vascular pattern suggesting it was growing some form of baleen-like tissue between its teeth, or behind its teeth, or along the posterior portions of its palate. Early, small, inefficient, but functional enough to be maintained by selection. The animal was using both strategies simultaneously. It could grab large prey with its teeth when the opportunity arose, and it could filter small prey with its proto-baleen when the teeth were not in use. Neither strategy was being done as well as a specialist would do it, but the combination was viable. This mosaic state persisted in various mysticete lineages for something like 5 or 10 million years, during which the balance between teeth and baleen gradually shifted. In some lineages, the teeth became smaller and less important, while the baleen expanded and became more central to feeding. In other lineages, the opposite may have happened temporarily. Evolution does not move in a single direction, even within a single clade. Different populations in different environments pursued different balances of the two feeding strategies, and the fossil record shows a range of intermediate forms rather than a clean linear progression. Eventually, in the ancestors of modern baleen whales, teeth disappeared entirely. The last functional adult teeth were lost somewhere around 25 million years ago, and from that point forward, the Mysticetes lineage was committed to filter feeding. A remarkable fossil discovered in 2018 showed that the transition was actually more complex than a simple replacement of teeth by baleen. The animal, Maiacetus nesbiti, lived in what is now Oregon about 33 million years ago. It is a stem Mysticetes, closer to the ancestral Mysticetes condition than modern baleen whales, but past the split with Odontocetes. And the thing that made Maiacetus interesting, when Carlos Peredo and his colleagues described it, was that it had neither teeth nor baleen. The skull shows no evidence of functional teeth. The tooth sockets have regressed, but it also shows no evidence of baleen. The palatal foramina that would be expected if baleen were present are absent. This animal was apparently a suction feeder. It used its toothless mouth to create pressure differentials that pulled small prey into the oral cavity, something like how many beaked whales still feed today. The evolutionary implication is that teeth were lost before baleen was gained. There was an intermediate stage, lasting perhaps a few million years, in which Mysticetes had lost their teeth but had not yet evolved baleen, and they made their living by a different feeding strategy entirely during that interval. This refines the standard story considerably. The old picture, teeth turning into baleen in a direct sequence, was replaced, in light of Maiacetus, with a more complex picture in which tooth loss and baleen evolution are decoupled events, separated by an evolutionary stage of suction feeding. Each stage was viable on its own. Each stage had a different way of making a living. The lineage moved through these stages not because it was heading somewhere specific, but because each stage's descendants happened to face conditions where modified versions of the feeding apparatus were selected for, and the cumulative modifications eventually produced the baleen whales we know. The molecular side of this story is, if anything, even more striking. The genes required to build mammalian teeth, specifically the genes that encode the proteins of tooth enamel, including enamelin, ameloblastin, amelogenin, and matrix metallopeptidase 20, are still present in the genomes of every living Mysticetes, but they have been inactivated. The inactivations took the form of various mutations: stop codons inserted in the middle of coding sequences, frame-shift mutations that scrambled the reading frame, and in one particularly notable case, the insertion of a Cene retrotransposon into the middle of an exon of MMP20, disabling the gene entirely. These mutations occurred during the evolutionary history of Mysticetes at different times, accumulating across millions of years as the selective pressure to maintain functional enamel genes disappeared. The genes are still sequenced, still present in the same locations of the genome, still bearing the structural signatures of genes that once worked. They are pseudogenes, genetic fossils, as it were, embedded directly in the DNA of every living baleen whale. This is a different kind of vestige than the Basilosaurus legs we looked at several chapters ago. The legs were an anatomical residue, visible in the bones of the animal, reduced but still present. The enamel genes are a molecular residue, invisible at the level of the whole organism, but readable in the genome if you know how to sequence it. Both are signatures of the same kind of evolutionary phenomenon, the persistence of structures or sequences whose original function has disappeared, maintained in reduced or inactivated form because the cost of removing them is not obviously smaller than the cost of keeping them. Your own genome contains equivalents. You carry broken olfactory receptor genes, about 800 of them, that were functional in your rodent and primitive primate ancestors but became pseudogenized as your lineage shifted toward visual dominance and reduced reliance on smell. You carry the remnants of genes for yolk proteins, active in your reptilian ancestors, broken in the common ancestor of placental mammals when placental nutrition replaced yolk-based embryonic support. You carry, embedded in your DNA, the genetic record of what your lineage used to be, the same way every mysticat carries the record of its toothed ancestry. The living baleen whale is a body shaped, at every level from embryo to adult to genome, by its evolutionary past. The fetus briefly grows teeth it will never use. The pseudogenes for enamel sit inactivated in every cell. The palatal vasculature that once supplied nothing more than the soft tissue of the gums now supplies the baleen plates that replace them. And when the whale swims, mouth open, through a dense patch of krill or small fish, filtering prey from several tons of seawater in a single gulp, it is doing something that no toothed ancestor could have done and that the lineage had to lose its teeth to learn. The loss made the gain possible. Evolution does not run in straight lines. It does not optimize, it substitutes. What makes a baleen whale possible is not the addition of baleen to an otherwise unchanged body. It is the removal of something old to make room for something new, repeated at many levels across many structures until the resulting animal has no obvious resemblance to its own ancestors. There is a kind of whale that, if you follow its life closely enough, seems to spend most of its existence doing something impossible. A Cuvier's beaked whale in the open waters off the California coast or in the deep basins of the Mediterranean will surface to breathe for perhaps 5 or 10 minutes, filling its lungs several times in quick succession, and then it will close its blowhole and begin descending into water where the pressure and the cold and the darkness conspire to make most vertebrate life unsustainable. Over the course of the next 2 hours, sometimes longer, it will reach depths below 2 kilometers, hunt squid and fish in absolute darkness, produce and process echolocation clicks at frequencies well above anything a human can hear, maintain brain function under pressures that would collapse a human's lungs, and then return to the surface, exhale, inhale, and begin again. The world record for a mammalian dive set by a Cuvier's beaked whale tagged off the coast of California in 2014 is 3 hours and 42 minutes at a depth exceeding 2,990 meters. Nothing else in the mammalian lineage even approaches this. The animal has spent an evolutionary path making something that should not be possible into something routine. The sperm whale is similar, though its numbers are slightly less extreme. Sperm whales routinely dive to 1,500 meters, regularly reach 2,000, and the maximum confirmed dive is approximately 2,250 meters, with unconfirmed reports suggesting dives approaching 3 kilometers. Dives of 60 to 90 minutes are typical. Between surfacing intervals, a sperm whale spends a minority of its life in air. The majority is spent beneath it, hunting, traveling, resting, socializing at depths where the physics of the environment would kill almost anything else with a backbone. How any of this works is one of the most thoroughly studied problems in comparative physiology, and the answer involves no single dramatic adaptation, but rather a coordinated suite of modifications, most of which are elaborations of capabilities that every mammal already has. This is worth emphasizing at the outset because the popular picture of deep-diving whales sometimes suggests that they have evolved some kind of exotic physiology unavailable to other mammals. The truth is closer to the opposite. The diving whale's physiology is the ordinary mammalian physiology pushed and pushed for tens of millions of years against the limits of what the underlying mammalian body plan will tolerate until capabilities that would be impossible for a human have become routine for a whale. Begin with oxygen storage. Your body stores oxygen in 3 places: in your lungs, in your blood bound to hemoglobin, and in your muscles bound to myoglobin. The relative contributions vary by species, but in humans, the lung store is the largest, the blood store is second, and the muscle store is relatively small. In deep-diving whales, the distribution is flipped. The lung store, counterintuitively, is proportionally smaller than in terrestrial mammals because inflated lungs under pressure are a liability at depth. The compressed air causes problems with nitrogen absorption and decompression. Most of a diving whale's oxygen is stored in its blood and its muscles. Blood volume, relative to body weight, is much higher in diving cetaceans than in land mammals, and the muscle oxygen storage, driven by extraordinarily high concentrations of myoglobin, is in a different league entirely. This is where the evolutionary story becomes interesting, because the biology of cranking up myoglobin concentrations in muscle is not straightforward. Myoglobin is a small protein that binds oxygen in the same basic way hemoglobin does, holding it in reserve in muscle tissue for use during periods of activity or oxygen deprivation. Your own muscles contain myoglobin at concentrations of around 1 to 3 milligrams per gram of tissue. A seal has 10 to 30 times more than that. A sperm whale has up to 50 times more. And the reason mammals can't just produce ever higher concentrations of myoglobin up to whatever level their diving lifestyle would benefit from is that myoglobin molecules at high concentrations tend to stick to each other. They aggregate. They precipitate out of solution. The cellular machinery breaks down if you try to load too much of the protein into a given volume of muscle. The solution that deep-diving mammals have arrived at, independently in multiple lineages, is a chemical modification of the myoglobin protein itself. In a 2013 paper published in Science, Scott Maiacetus and his colleagues at the University of Liverpool reconstructed the evolutionary history of myoglobin across about 130 mammalian species using ancestral sequence techniques that allow researchers to infer what the protein looked like at various points in the past. What they found was that diving mammals across multiple independent lineages, cetaceans, seals, sea otters, beavers, even some of the ancient semi-aquatic precursors, had accumulated amino acid substitutions that increased the net positive surface charge of the myoglobin molecule. The increased positive charge makes the molecules electrostatically repel each other at high concentrations, preventing aggregation, allowing myoglobin to be packed into muscle at levels that would be chemically impossible with an unmodified protein. The same adaptation for the same physical reason in several unrelated lineages of diving mammals. Another case of convergence, this time at the molecular level, in a specific protein with specific amino acid changes at specific positions on the molecular surface. The whales, in particular, have taken this further than any other mammalian lineage. A sperm whale swimming toward the bottom of the Pacific has myoglobin concentrations in its dive muscles that would be lethal in a land mammal. The protein would aggregate and destroy the cell. The whale's modified myoglobin, with its unusually high net positive charge, stays in solution. The oxygen it binds is available to sustain the whale's muscle activity for the 90 minutes of the dive. When the whale surfaces and breathes, the myoglobin is recharged. Nothing about this system is new. Every mammal has myoglobin, every mammal has oxygen-storing muscle tissue, every mammal has the same basic chemistry of iron-based oxygen binding. The whale has simply taken the standard mammalian toolkit and pushed it much further than any land mammal can afford to. Alongside the oxygen storage adaptations are a set of circulatory adjustments that kick in the moment a dive begins. Your heart rate, when you hold your breath and submerge your face in cold water, drops. That is the mammalian diving reflex we discussed in the introduction to this documentary, and you carry it in reduced form as part of your basic mammalian inheritance. In a sperm whale, the same reflex is extended dramatically. Heart rate drops from the surface swimming rate of something like 10 or 15 beats per minute down to 4 or 5 beats per minute during deep dives. Blood flow is redirected away from peripheral tissues, the skin, the extremities, the muscles that are not immediately needed, and concentrated on the heart, the brain, and the central nervous system. This is called peripheral vasoconstriction, and it means that during a long dive, most of the whale's body is operating on minimal oxygen, while the tissues that cannot tolerate oxygen starvation, the brain in particular, continue to receive adequate supply. The lungs themselves, rather than being inflated at depth the way a human's would be, collapse. Deep-diving whales exhale most of the air in their lungs before beginning a dive, and what little air remains is compressed by the increasing pressure until the alveoli, the tiny air exchange sacs in the lungs, collapse entirely. The whale descends with essentially no gas exchange happening in the lungs at all. The oxygen it uses during the dive is the oxygen stored in its blood and its muscles, not the oxygen in its lungs, because there isn't any meaningful volume of lung air to draw from. This prevents a whole class of problems that would otherwise make deep diving impossible. Nitrogen at high pressure dissolves into the blood. When an animal surfaces, the dissolved nitrogen bubbles out, causing what in humans is called decompression sickness, or the bends. Whales avoid this by simply not having significant gas-exchanging volume in the lungs during the deep part of the dive. The collapse of the alveoli, counterintuitively, is what makes the dive survivable. All of these adaptations are present in weaker form in every mammal. Your own diving reflex is real. You can test it by immersing your face in cold water and monitoring your pulse. But it works at a level that barely registers compared to a whale's. Your myoglobin is structurally similar to a whale's, differing mainly in the surface charge modifications that let whales pack much more of it into their muscles. Your lungs would collapse under sufficient pressure, though the pressures involved would kill you long before that happened. Your peripheral vasoconstriction during cold exposure is the same mechanism, in weaker form, that concentrates blood flow to central organs during a sperm whale's dive. The mammalian toolkit for dealing with oxygen deprivation and water immersion is ancient, shared and present in every mammal, including humans. The whales have taken each element and pushed it to its physiological extreme. But the elements themselves are yours too, running in reduced form every time you hold your breath or step into cold water. There is one more physiological feature worth mentioning because it connects the diving adaptations back to the body archive framework we have been using throughout this documentary. Your spleen, sitting in the upper left quadrant of your abdomen, is an organ you probably do not think about much. It functions in blood filtration, in immune response, and, relevant here, in red blood cell storage. In a resting mammal, the spleen contains a significant reserve of red blood cells that are not in active circulation. Under conditions of acute oxygen demand, vigorous exercise, blood loss, breath holding, the spleen contracts, expelling its reserve into the circulating bloodstream and increasing the blood's oxygen-carrying capacity by several percentage points. In deep-diving whales, the spleen is proportionally much larger than in terrestrial mammals, and its red blood cell reserves are correspondingly larger. During a dive, the whale's spleen contracts, dumping additional oxygen-loaded red cells into circulation, extending the dive's aerobic budget by a measurable amount. The same organ doing the same job at different scales. When you exercise hard enough to feel your heart pound and your breath come quick, your spleen is contracting. You can sometimes feel the side stitch pain that accompanies this, usually on the left side of the abdomen during running, the feeling of your spleen expelling its red blood cell reserve to keep up with the muscle demand. This is the same mechanism a sperm whale uses at 2 kilometers depth. The organ is yours, the function is yours. The whale has optimized it. You have it in quieter form, and you use it every time you push your body hard enough to matter. The sum of all these modifications is what allows a Cuvier's beaked whale to spend 3.5 hours beneath the surface, or a sperm whale to hunt squid in the dark at 2 kilometers down. None of it is new. All of it is the ordinary mammalian body run at its extreme. The genome of a diving whale contains essentially the same genes as the genome of a land mammal: myoglobin, hemoglobin, spleen-regulating transcription factors, cardiovascular control pathways, and the differences are matters of tuning rather than invention. Selection over 50 million years has pushed each parameter further than any terrestrial mammal ever pushed it, but it has not built anything from scratch. The whale at depth is the mammal at depth. Its capability is yours, extended into a regime you cannot visit and could not survive. Somewhere beneath the surface tonight, one of them is diving. Roughly 4 million years ago, during what paleoceanographers call the late Pliocene, the global ocean circulation began to shift in a way that would change the character of marine ecosystems across the entire planet. The closing of the Isthmus of Panama, which had been a gradual process throughout the preceding several million years, was finally complete. The previously open connection between the Atlantic and the Pacific near Central America had sealed itself off, and the consequences for global ocean currents were substantial. The Gulf Stream intensified, the thermohaline circulation reorganized. Surface winds over the major ocean basins shifted in patterns that, over the following million years, produced increasingly strong seasonal upwelling along continental margins. The periodic rise of cold, nutrient-rich deep water to the surface in specific zones, driven by wind-blown displacement of surface waters away from the coasts. This is a more important development than it might sound. Upwelling is one of the most important biological phenomena in the oceans, because the deep water it brings to the surface is rich in the dissolved nutrients that phytoplankton need to grow. When upwelling is strong and seasonal, entire coastal regions become enormously productive during the upwelling months, producing phytoplankton blooms dense enough to turn the water green, supporting zooplankton populations that explode in response, and creating patches of marine productivity that concentrate small organisms into dense aggregations over specific times and places. The modern California current, the Humboldt current off Peru, the Benguela current off southwestern Africa, and the Canary current off northwestern Africa are all major upwelling zones, and they are all among the most productive marine ecosystems on Earth. Before the late Pliocene, upwelling was present but weaker and less seasonal. After the late Pliocene, upwelling became the ecological engine driving a substantial fraction of ocean productivity worldwide. For filter-feeding whales, this was transformative. The baleen whale feeding strategy, lunging through dense patches of small prey, filtering huge volumes of water through baleen plates, extracting the prey as the water passes through, works optimally when prey is concentrated in discrete patches rather than distributed uniformly through the water column. Pre-Pliocene oceans tended to have more uniformly distributed prey. Post-Pliocene oceans, with their intensified upwelling systems, produced the kind of dense, patchy, seasonal prey aggregations that lunge feeding requires. Mystic-seat whales that could travel between these productive zones, feed intensively during bloom seasons, and store the resulting energy as blubber for leaner months, had access to a food supply that had essentially not existed before. And the body plan that exploits this food supply most efficiently, as it turned out, is the extremely large body plan. This is counterintuitive. We tend to assume that large body size confers its advantages on land, the capacity to dominate competitors, to travel long distances, to intimidate predators. In the water, the calculus is different. Large filter feeders benefit from body size for specific energetic reasons. The volume of water a whale can engulf in a single lunge scales roughly with the cube of its body length, while the metabolic cost of that lunge scales roughly with the square of its length. Bigger whales, in other words, can filter disproportionately more water per unit of effort than smaller whales. They can also store more blubber, which means they can survive longer between feeding seasons and travel further between productive zones. And they can dive longer on a single breath, because larger bodies have proportionally more oxygen storage capacity relative to their metabolic demand. Every one of these advantages reinforces the others. Once the ecological conditions favored body size, the selection pressure pushing mysticetes toward larger sizes was sustained, substantial and consistent across multiple independent lineages. The result, documented in detail by Graham Slater, Jeremy Goldbogen and Nicholas Pyenson in a 2017 paper in the Proceedings of the Royal Society B, is that the extremely large body sizes we associate with modern baleen whales are a remarkably recent phenomenon. For most of mysticete evolutionary history, from the Oligocene origin of the group through most of the Miocene, mysticetes were moderately sized animals, typically between 5 and 15 meters long. The truly enormous sizes, the sort of sizes represented by the modern blue whale, emerged only within the last 3 to 4 million years. Multiple independent mysticete lineages underwent rapid size increases during this window, converging on the giant body plan from different starting points. The blue whale, the fin whale, the bowhead, the right whales, each of these lineages increased in body size substantially during the Pleistocene, and the simultaneity of the increases across unrelated mysticete lineages points to a shared environmental driver. The driver, the analysis concluded, was the intensification of upwelling and the consequent availability of dense, seasonal, patchy prey. A subsequent discovery, published in 2019 by Felix Marx and his colleagues, complicated this picture somewhat. The authors described a blue whale fossil from southern Italy, approximately 1.5 million years old, that reached lengths of up to 26 meters, close to modern blue whale sizes, but at a date slightly earlier than the 2017 analysis had suggested. The gigantism, in other words, may have arrived somewhat earlier than the Pleistocene estimate, pushing the origin back into the late Pliocene, or perhaps slightly before. The underlying story remains the same. These are geologically recent animals produced by selective pressures that became strong only in the last several million years during the ocean reorganizations of the late Cenozoic. But the exact timing continues to be refined as new specimens come to light, and the active debate in the literature is over whether the true origin of modern gigantism is 3 million years old or closer to 5. The blue whale itself is worth sitting with for a moment because it is the most extreme result of this evolutionary process, and because, in certain quiet ways, it connects directly to your own body. An adult blue whale can reach 30 meters in length and can weigh up to roughly 180 metric tons. Its heart, if you could hold it, would weigh approximately 180 kilograms, about the weight of 3 average adult humans. With each beat, that heart pumps roughly 220 liters of blood through the animal's circulatory system. Its aorta, the main artery leaving the heart, has an inner diameter of something like 20 to 30 centimeters, large enough at its widest for a small adult human to crawl through if there were any reason to. The tongue alone in an adult blue whale can weigh as much as an elephant. The animal's low-frequency vocalizations can be detected by sensitive hydrophones across hundreds of kilometers of open ocean. Every one of these numbers represents a scaling up of the same mammalian architecture you carry in your own body. Your heart weighs approximately 300 grams and pumps approximately 70 milliliters of blood per beat. The blue whale's heart weighs roughly 600 times what yours does and pumps roughly 3,000 times the volume, but the heart itself, structurally and functionally, is the same organ. 4 chambers, 2 atria, 2 ventricles, valves between them, a muscular pump driving oxygenated blood through an arterial tree and collecting deoxygenated blood through a venous tree. The architecture that supports your circulation tonight, as you're listening to this, is the same architecture that supports a blue whale's circulation at scale. The valves work the same way. The contraction sequence is the same. The electrical conduction system that coordinates the heartbeat is the same underlying pathway. Selection has scaled the mammalian heart across the Maiacetus lineage by a factor of several hundred without changing anything fundamental about how it works. Nothing new had to be invented. The template was already there, in your ancestors and in the blue whales, running for tens of millions of years before the Maiacetus lineage began enlarging it. There is an embedded observation here about what blue whale size actually is, philosophically. We tend to think of enormous animals as a kind of evolutionary achievement, as the lineage in question having accomplished something that lesser lineages couldn't manage. But the blue whale's size is not an achievement. It is a consequence of sustained selective pressure in a specific ecological regime that only became available in the form it now takes within the last few million years. If the Isthmus of Panama had not closed when it did, if the late Cenozoic ocean circulation had not reorganized in the way it did, if upwelling patterns had remained at their pre-Pliocene intensities, the blue whale would not exist. The lineage would have had no particular reason to scale up to that size. A 15-meter mysticete, fed by pre-Pliocene marine productivity, would have remained a 15-meter mysticete. The largest animal ever to live on this planet is, in an important sense, the product of a relatively recent and contingent geological accident, not a long trajectory of evolutionary ambition. Something similar is true of the lunge feeding system itself. The specific feeding mechanism that allows a rorqual whale, the group that includes the blue whale, the fin, the humpback, and several others, to engulf several tons of water in a single lunge relies on an integrated set of anatomical features that have been studied in recent decades in considerable detail. The jaws are separate from each other at the front rather than fused, allowing enormous mouth gapes. The throat pouch, called the ventral groove blubber, expands dramatically during a lunge, accordion style, inflating to many times its resting volume. The tongue inverts during the engulfment, becoming part of the expanding pouch rather than occupying space in the mouth. And, as a 2012 paper in Nature by Nicholas Pyenson and his colleagues documented, there is a specialized sensory organ embedded in the chin between the two unfused jaws, a cluster of mechanoreceptors in a gel-like matrix, whose job is to register the expansion of the throat pouch during the lunge and coordinate the timing of the jaw closure and the water filtration. This organ was not known to exist before 2012. Its discovery solved a long-standing puzzle about how rorquals coordinate their lunge feeding, and it confirmed that the lunge feeding system is a genuinely integrated mechanism with all its components evolved together, rather than a collection of separately selected features that happen to work in combination. The rorqual lunge is the blue whale's defining act, the thing that lets it consume up to 4 tons of krill in a single day during feeding season. And it is a behavior that no land mammal and no pre-Pleistocene whale could have executed. It required the ocean productivity conditions, the body size, the anatomical integration, the sensory coordination, all of which came together in the Maiacetus lineage only within the last several million years. The blue whale, in other words, is younger as a specific evolutionary entity than most people imagine. The lineage is old. The group is old. The specific body and feeding mode that defines the modern blue whale is geologically young, produced by a recent ocean and a recent ecological opportunity, and still operating in conditions that have not yet had time to change around it. Late Pliocene oceans are cold now. Antarctic sea ice has been expanding seasonally for millions of years. The krill populations that sustain the blue whale are concentrated in polar and subpolar waters, dense enough to support the largest animals the planet has ever produced, seasonal enough to demand the long migrations these whales still undertake. Somewhere tonight, in the Southern Ocean or the northern North Pacific or the deep North Atlantic, a blue whale is moving through water so cold that even its specialized physiology has to work to maintain body temperature, breathing at the surface between long periods below it, carrying a heart the size of a small car through a body that has no immediate equivalent in the evolutionary record. It is not the last of anything. The lineage continues. The size it has reached is the size the current ocean selects for, and when the ocean eventually changes, which it will over geological time, the size will change with it. Somewhere in the Southern Ocean tonight, below a layer of pack ice or through open water between the ice shelves, a blue whale is breathing. The intervals between breaths are long, measured in minutes rather than seconds, and between them the animal is somewhere beneath the surface, working its way through a dense patch of Antarctic krill or traveling slowly toward one. The water is cold, perhaps a degree above freezing, and the darkness under the surface is almost absolute because there is no moon tonight over that part of the ocean. The whale's heart, beating at perhaps 4 or 5 beats per minute during the deepest part of its descent, is pumping blood through a body that stretches nearly the length of a tennis court. Its vocalizations, at frequencies just below the threshold of human hearing, are propagating outward through the water in pulses that will still be detectable by sensitive hydrophones hundreds of kilometers away. It is doing nothing remarkable. It is doing what its ancestors have been doing in one form or another for about 50 million years. And the thread that connects this animal to the first of its ancestors has never, at any point, been interrupted. The story we have been telling across the last several hours has a shape to it, and the shape is worth summarizing briefly before we let it go. A lineage of small terrestrial mammals living at the edges of a warm, shallow sea about 50 million years ago began to modify certain features of their bodies in ways that made them slightly better at exploiting aquatic prey. One specific feature, the bony architecture surrounding the inner ear, changed first, and it changed because directional underwater hearing gave individuals who possessed it a measurable advantage over individuals who did not. The rest of the body followed slowly across millions of generations. Legs reduced and eventually disappeared from external view. Nostrils migrated up the skull and became a blowhole. Teeth in one lineage were lost and replaced by keratinous filter plates. A whole suite of physiological systems, circulatory, pulmonary, sensory, were pushed to accommodate longer dives, deeper waters, colder temperatures, and larger body sizes than any terrestrial mammal had ever needed to tolerate. The end point of this process, as of tonight, is a set of living species that range from 6-foot vaquita porpoises to 30-meter blue whales, occupying essentially every marine habitat on Earth, linked to their Eocene ancestors by an unbroken chain of parent-to-offspring inheritance that has been running continuously for about 2 million generations. Nothing planned this, nothing aimed at it. The animals that produced the whales were not trying to produce whales. Pakicetus was just hungry. Indohyus was just trying to hide from predators. Basilosaurus was just living its own predatory life in its own warm Eocene sea. Each of these species was, in its own moment, the current endpoint of its own lineage's history, with no knowledge of what would come after it and no mechanism for that knowledge to matter, even if it had existed. The extraordinary fact of what the lineage eventually became is visible only in retrospect, from the vantage of an observer who can see the whole ark from outside. Inside the ark, at any given moment, there was only a population of animals doing what its local conditions rewarded. This is the thing that evolutionary biology keeps trying, against the grain of ordinary human cognition, to communicate. The elegance of the result is not evidence of design. It is evidence of what a blind statistical process can produce, given enough time, enough organisms, and consistent selective pressure. 50 million years is a long time. The number of individual whale and proto-whale lives across that span, if you could count them, would be some immense number, tens of billions, perhaps more. Each life a single roll of the genetic dice, each death a filter removing some rolls and allowing others to continue. The cumulative effect of that many rolls, under that much sustained selective pressure, is the blue whale breathing tonight off the coast of Antarctica, and the sperm whale clicking at 2 kilometers depth in the Pacific, and the humpback singing in Hawaiian water, and the beluga pushing through melting summer ice in the Arctic. Each of these animals is the current terminus of a thread that has never broken. You are also the current terminus of a thread. The thread that leads to you is longer than the one that leads to the blue whale. It runs back through your primate ancestors to a common mammalian ancestor shared with all living mammals, and from there to the first mammal, and from there to the synapsid reptiles of the Permian, and from there to the first tetrapods crawling out of Devonian swamps, and from there to the lobe-finned fish that preceded them, and from there back through every vertebrate and every chordate and every eukaryote, all the way to the first self-replicating molecule on a young Earth roughly 3,800,000,000 years ago. Every ancestor on that line, without exception, survived long enough to reproduce. This is not a remarkable fact. It is the only fact compatible with your existence. If any single one of your direct ancestors, the parent mammal, the grandparent mammal, any of the billions of ancestors stretching back into deep time, had died before reproducing, the entire chain of descent that produced you would have been broken, and you would not be here to consider it. Your thread and the blue whale's thread share most of their length. The two threads diverge somewhere in the early Paleocene, maybe 70 million years ago, before anything resembling a whale had begun to exist. Before that point, your lineage and the cetacean lineage ran together, through the same ancestors, the same populations, the same generations of small mammals living in the same forests and fields and coastlines. For something like 3 billion years of evolutionary history, the threads were one thread. The divergence happened only recently by the standards of the whole arc. For the first 95% of the time since life began, your ancestors and the blue whale's ancestors were the same organisms. This is a kind of kinship that does not have an ordinary name. You and the blue whale are not cousins in the colloquial sense. You cannot trace a simple family tree to a recent common grandparent. But at the level of the whole biosphere, across the scale of evolutionary time, you are siblings of the same ancient lineage. You share a common ancestor with every mammal, every vertebrate, every animal, every eukaryote on Earth. The things that distinguish you from a blue whale are recent modifications to a shared mammalian template. The things that distinguish you from a squid are recent modifications to a shared animal template. The things that distinguish you from a mushroom are recent modifications to a shared eukaryotic template. At the deepest level, the thread is the same thread and the branches are all continuing. There is something I want to let land here if you are still with me. The story of the whale is often told as a story of transformation, and that is true, but it is not the whole truth. It is also a story of continuation. Nothing about the whale is disconnected from its ancestors. Nothing about its body is entirely new. The four-chambered heart that pumps its blood is the heart every mammal has. The three middle ear bones that receive acoustic signals are the bones every mammal has. The spleen that contracts to release red blood cells during a dive is the spleen every mammal has. The genetic code that specifies its proteins is the same code every organism on Earth has been using, with minor variations, for several billion years. Selection has modified the emphases, shifted the proportions, pushed certain capabilities further than any land mammal would push them. But the whale has not escaped its mammalian inheritance. It has extended it. The blue whale is a mammal, doing what mammals do at a scale and in an environment that makes the ordinary mammalian business look extraordinary. What this story is, underneath the specific biological details, is a particular way of understanding your own place on the planet. You are not a designed being. You are a statistical residue of a 3.8 billion year process that had no aim, no plan, no designer, and no preferred outcome. The process happened to produce you as one of its many current endpoints, and it happened to produce blue whales and sperm whales and humpbacks and every other living species as other current endpoints. None of these endpoints is more advanced or more evolved or more successful than any of the others. They are all, equally, the current terminus of their own threads. The octopus watching from a tide pool is as evolved as you are. The lichen crusting an Arctic rock is as evolved as you are. The mycelium running through the forest floor beneath your feet is as evolved as you are. Each of these is the latest version of a lineage that has been running without interruption for as long as yours has. And each of these is carrying in its own body the record of everything its lineage has been. The whale carries its toothed ancestors in its embryonic tooth buds, its terrestrial ancestors in its vestigial pelvic bones, its Pakiceted ancestors in the bony architecture of its ear. You carry your mammalian ancestors in your four-chambered heart, your tetrapod ancestors in the bones of your limbs, your chordate ancestors in the rod-like cellular structure that organized your embryonic body, your prokaryotic ancestors in the mitochondria that power every cell in you. You are not just yourself. You are also every organism that preceded you, present in compressed form, woven into the architecture that you walk around in and that you are going to sleep in tonight. The thread continues. As you are listening to this, somewhere on Earth, a whale is breathing at the surface of the ocean. Its breath carries, dissolved in the air that fills its lungs, and will fill them again at the next inhalation, the same oxygen that is in the room with you. The oxygen molecules passing through your lungs tonight have been through the lungs of a whale somewhere at some recent point. The water circulating in your bloodstream has cycled through the world's oceans many times. The carbon in your body was in the bodies of countless other organisms before it was in yours, and will be in countless others after you are gone. You are not separate from any of it. You never have been. The story that began 50 million years ago at the edge of a Pakistani river with a small hoofed predator rebuilding its inner ear to hunt in shallow water is the same story that has arrived at a blue whale breathing under the Antarctic tonight, and at you, in whatever room you are in, hearing these words. The lineage continues. Your own lineage continues. The threads, all of them, are still being spun. And somewhere at the other end of one of them, a mammal the length of a tennis court is exhaling slowly into a cold, black sea, and then inhaling, and then going back down into the dark. Rest now. You are safe. The thread will keep running without you watching it. Sleep well.